Okay, so the idea was that can we do something about it, like what he says, Mahavani Lavavi. So I tried to do something at NCL with the help of my colleagues, and we identified around 100 plus plants based on the northern Western Ghat, and we studied their association. That means, you know, the way, like, so for example, you would like to sit next to your friend or next to somebody who like, and you don't want to sit somebody who you don't like. So plant also have similar nature. So if you study the association of the plants in the nature, not in our agriculture crop plants, by the way, if you ask this question to the like, nature, and if you see there is a specific arrangement which very much exists there. So we studied that and we come up with a plan to grow 110 plants in around 10 gunta land. That means 10,000 square feet. And we were very enough lucky that time. Our science and technology minister, Dr. Harshvardhan ji, as well as our DG, Dr. Shekhar Mande, Professor Ashwini Kumar Nangia. There are people from Central University of Hyderabad. They will be happy to see. So all of us started this small effort to make it like, can we really follow what Sant Jnaneshwara says, Mahavani Lavavi? And we were so happy about it that again, I think, yeah. So we started growing this plant in the area which was really in a bad shape. What happened is within 1 year, that plants came. And all of you know, if you are working on a student of biology or zoology, you know that there are some insects which feed on a specific plant, and there are some insects which feed on multiple plants. So what was very surprising for us is we were expecting that there will be a lot of insect came. So this is a survey done by this PhD coursework student in a 1 square meter areas of that forest versus other areas in the NCL. And they found there are no insects in other area, but wherever we planted these 110 plants, there were several insects were visiting. And the beautiful part of this was that the insects which feeds on multiple plants, you can expect that they come. But then we planted only one plant of a specific species. And then the insects which are very specific for that plant, which is called monophagus, our specialist, they were also there. And then we couldn't understand, and we were like surprised how this one plant that insect has identified. And that's what I feel the basis of identification is metabolites, volatiles, the chemicals that plant produces. So who said that plant don't talk? Plant do talk. Okay, they don't talk Marathi, Hindi, or Telugu, or English, or whatever other language. They talk a chemical language, and that we don't have to really worry whether you are in Maharashtra or Andhra or any other part of the world. This language remains the same. Chemical language remains the same. And one classical example is, all of you know, the pheromones, for example. They are so unique to the specific insect and specific plant or specific male-female interaction. So this chemical language is something very beautiful. And I think we are trying to understand the language of the plants or language of the nature. I also want to tell you, I also attended a talk from morning, and of course, wonderful talks and so on. But what I feel is when I keep talking to the students and even the researchers, most of our questions, not necessarily in the life sciences, but most of our questions are human-centric. That means we have to develop something to increase our lifespan. We have to do something to avoid cancer or diabetes or so. I feel as a biology student, we should ask the question nature-centric. Because if nature lives, then only we live. Even if we are not selfish, in my opinion. And why today we are worried about so much of climate change and so on? It's not because of that. Because we feel that the first something that will get away from this motherland is human beings. So we should ask, as a biology student, I appeal all of you that you should ask the question which are nature-centric, not human-centric. We come here, stay for some time, maybe have a life of around a good life around 30-40 years, and overall life maybe 60-70 years. But then we need to think about nature. And if you ask this question, because why I'm saying, sir, this, because I take a classes for PhD coursework, and I study, ask them, I study or I teach some kind of plant insect interactions, and I ask them which is the best system to study plant insect interaction, whether native plants and their insects, or we should study in the crop plants. And most of the question comes that we should study in the crop plants or agriculture field. And I said, why? Because they say that pest is a major problem. That means insects are enjoying the plants. If I study that, what I am going to learn? Nothing. If you study something in the nature, like the plants which are growing outside, you don't spray them, you don't use fertilizers, you don't irrigate them, but they are surviving. So something, if you want to discover or understand or actually identify novel molecules or novel mechanisms, we should study where the mechanisms are there. In Marathi, it is said, "Adat asil तर पोवऱ्यात येईल ना." So if these crop plants are highly susceptible, insects are enjoying it, and they are like damaged by the insects. So if you study that, what we are going to gain? So these are some of the questions actually are important when you think about a nature-centric view and not human-centric view. So this also, it took some time for me to understand this. Like, not during my PhD, I did not get opportunity to attend such a conference. You guys are so lucky. Your faculties are so good, sir. They are organizing this conference almost every year, and all the students get opportunity to attend one or two conferences during their masters, which is really, really good. So this kind of approach, it took me some time even after PhD to understand, if I'm working on plant insect interaction, I should not really think about the insect which makes damage to the plant. We should think, and all of you know that pollinators are also important. They are also insect, but it is beyond that. It doesn't restrict to only pollinators and insect pests. If you think about something like this carnivore, there is a tritrophic interaction. That means if this plant tries to defend and if it cannot manage, it secretes some volatiles that attracts the enemy of enemy. And that's what called tritrophic interaction. And a lot of other things happens even above ground and below ground. And not only that, it also has to compete with the neighboring plants. So that's the beauty of nature. That's the beauty of plant. And I quote this for you because human genome was sequenced around 2000. Yes, sir, around 2000. Whereas plant genome was sequenced after 10 years, that is 2010. And the first plant genome sequence came out from a model plant called Arabidopsis thaliana, which is actually this big plant, maybe not more than 1 feet. And its life cycle is around 2 to 3 months. So when we looked into the genome, what we identified in the human genome is there are something like 22 to 25,000 genes. You can correct me if I'm wrong. Whereas in Arabidopsis, a plant which lives only for 3 months, when they sequence this genome after 10 years, they found there are 45,000 genes. So then who is rich? We or plants? So why? When this question is why? Why plant have more number of genes? And that is true for metabolites also. Why? Because we studied from in our childhood that plants cannot move. If you feel too cold in this room or too hot in this room, you have option to go out. Plant don't have, do or die. So that's why they have much more flexibility at genetic level, at molecular level, at biochemical level, at enzyme level, and so on. And that's the beauty to study. I will say the plant and the mechanisms that exist there. So when you think about a plant and insect, and I'm talking in the context of bioactive molecules, because all this is happening because of bioactive molecules. They are not produced. That's what I'm saying, that you don't ask this question human-centric. Bioactive molecules we can use to treat diseases also, but bioactive molecules are very significant role within the plant. Plant don't have cancers. Why they should produce anti-cancer compounds? We can use them. That's perfectly all right. I'm not saying it's wrong, but plant do have their specific role for these molecules. So the first plant in the form of algae came on this Mother Earth around 440 million years ago. Something like 440 million years ago, whereas the first insect came on this Mother Earth around 400 million years ago. And these interaction between plants and insects are always there, and probably they were balanced service-resource relationship, but something happened. So you can be a friend, but at certain stage, your friend can become a threat for you. Yeah, this is, we know that in 3 Idiots, they show that if your friend gets topper, then you have a problem. Somebody else topper, it's all right. So same thing. So we are also friend, but also we are sometimes don't like that our friend progresses. So probably it's something that happened between this plant insect interaction, and that becomes a molecular arm race. That means molecular war. That means plant is producing something to defend, and insect is trying to actually inactivate that defense and overcome. And that is what called co-evolution. That means plant produces something, then on the to encounter or to manage that, insect also produces something. And that is what we are trying to understand, this language. And if you can understand some of this, we can think about application, because I'm a strong believer that there is nothing like applied science or basic science or fundamental science and I will say technology. So Louis Pasteur, it's not me, Louis Pasteur said sometime back, there is science and application of science. Newton, if could have not put these laws, we cannot send satellite today. Yeah? He never thought that this laws will be used to send satellites. So don't differentiate. Science is science, and if you apply, any science has application. So it's actually time, sometimes time, and what is our need. So further, I don't want to go into the detail, but if you think of behavioral and morphological adaptation in the insect, even in the plants, there are several. All of you know, I don't want to go into the textbook knowledge, because there is something like insect nose, and all of you know why we called bondari or bollworm, because it feeds on specific, it doesn't feed on a whole plant. So they know how to avoid. So they don't want to eat leaf tissue. They will go to the tissue which is more important. For example, I like sweet. If the plate is in front of me, I will take first gulab jamun. You must have had a gulab jamun. It was nice. So avoidance is one of the things. That means they don't really eat which tissue is toxic or has more, I will say, problematic compounds, and so on and so forth. On the other hand, plant also produces a lot of things, and one of the things I'm sure you all know, all of you know cuticles, or I will cell wall, and so on. But more importantly, something called trichomes. And as a biology student, you should see the plants more carefully. Plant has small hairs on their leaf or even stem, and they are rich in the metabolites. So they put all bombs there, because you know, you don't put your army in Marathwada. We put army on our border. So if you imagine, the plants most exposed thing to the nature is their trichomes. Yeah, and that's how they try to avoid. So there is a lot of interesting work in overall in the world, but in our group also, we are working on the trichomes and how these metabolic pathways are actually very functional, very active in the trichomes, like hairy-like structures on the plant surfaces. And at molecular level, they know how to detoxify, how to sequestrize, how to really make it less toxic. On the other hand, plant do produces specialized metabolites or secondary metabolites, as well as protein like molecules, which can defend, which can help them to protect themselves against not only insects, but even pathogen and abiotic stresses, and so forth. So one of the direct defense which has been well studied is produce, for example, this is again a not tobacco plant that we use it to eat tobacco. This is a native plant which has, I will say, high density population in some of the states of America, like Utah and Arizona, which comes naturally in a high density. That means you can mimic a lot of things like what happens in agriculture, and it produces a lot of defense mechanisms, and that's how it really survives and sustains in that environment. So this same plant, for example, what I was saying, here you may see very interesting thing is like this fly is not actually eating anything. It is actually this caterpillar larva is made some infestation here, that it is a plant, and then plant actually tries to defend, but the defense is not so active, probably, or maybe not able to manage. So then it releases some volatile in the air and says that, yes, you come here, your host is here. And this fly is not really eating, but it is laying the egg on this larva. And as soon as this fly lays the egg on this larva, this insect's life cycle starts in this insect. This is called parasitoids, okay, natural enemy of this larva. And as soon as these eggs are laid by this fly, this larva stops feeding. It becomes sluggish because it's infestation. It's like the insect has another infestation, which is insect infestation. And then that's how it manages. And this is not only one mechanism. There are hundreds of such examples. I will show you next example, which may be more relevant after having a nice lunch. So you see, this is something, several of you, maybe the farmers' kids, yeah. So if you go to the field, you see that there are some plants which produces this nectary. And all of you know now sorghum plant is on the field, jawar is on the field. So we eat something like sakhar. It comes on that. So why this plant produces, and especially this is produced when there is an insect damage by these caterpillars. So why they produce such sugary molecules? So, you know, there's a reason. With this sugary molecule, these ants, mungle, they get attracted because they like. And these ants don't go directly to the sugar. First, they eat non-vegetarian food, like they eat this larvae. Once they finish, they go back to the desert, and then they have. So that's the natural mechanism again to attract the enemies of enemy. So instead of just fighting with enemy, you can call. We also do this. I mean, we also call enemy of enemy. If you cannot handle your enemy, we call enemy of enemy. So plant do this. Probably we learn from the nature only. Okay, so coming back to the this very interesting experiment one of the student did in my lab. She was working on osimum. One of the osimum species called osimum kilimancharicum, called camphor basil or caprutulus. And the other student, Rahul, was working on tomato. And they were growing the plants in glass house, transgenic glass house, what even Gajanan showed me. And she thought, or she realized that when she is growing the plant, her plants are not getting infested by the insect, whereas Rahul's plants were getting infested by the insect. So what she did is, without asking me, and that's why you should not worry about to your teachers that there is no facility. The good ideas don't need facilities. Good idea needs your panchendriyas. You have to keep your panchendriyas open. That means you have to observe, being a biology student, and then try to listen, try to talk, try to discuss with your friend. Don't feel that it is a secret. If something is secret, that means it doesn't have anything. That's what I feel. So if you discuss, you get a brighter ideas and a lot of inputs. So she planned one experiment without even asking me, and that's what happens in NCL most of the time. The students are more proactive, and I'm very frank, sir, to tell you that I learn more from my students than my gurus. They taught me something else, but in research, I learn more from my students. So she did an experiment, simple experiment. She put some tomato leaves on one side of the petri dish and another side osimum leaves, and released 5 larvae. And she observed for day 2, day 3, and day 4. And what was very interesting observation is that insect prefers to feed on tomato, not on osimum. This was the simplest observation without any hi-fi techniques and instrumentation. And then, of course, it was very interesting to see that data. So then later on, we did another 6 months work and identified and even patented the molecules from osimum, which are insect repellent. By the way, plants are actually, our nature's language is very simple. Nature's language is live and let others live. Our language is a problematic. We want it to live. That's our problem, and that's why we have a lot of issues by the nature. Then we characterize several molecules from this osimum, and 2 of them we found they are very nice, and we have right now some formulations, and probably it will be technology. This slide I'm specifically showing you. This is a sir last year, not last year, last month, or maybe in February. This paper published on cover page in Science issue. So what they have shown is that again, this is a native plant systems, not domesticated plant, not agricultural crop plants. They identified a new molecule to the science. This molecule was not known. It is called, long name, but it's a capriccyl putrescine green volatile compound, some hexanol. And this is a new molecule which is produced by the plant to manage against this leaf hopper. So there is a lot of new information which is there. We need to dig it out. And this molecule is too complicated. For example, it takes some substrate from this pathway called green GLV pathway, the green leaf volatile pathway, jasminoid signaling, which actually induces phenylpipenide biosynthetic pathway. So some molecules are coming from substrate, some intermediates are coming from this phenylpipenide pathway as well as polyamine pathway. And this is a very new molecule. All these 3 blue were new genes which were not known till February. Probably these researchers know probably a year before, but this is what the science. If you do in a very on a very native systems, or if you ask very right questions, that's what you should be able to discover. So coming back to this, because it's one of my favorite slides that I show, is that research till your MSc you can do in some subject, but then I think we are, at MSc level also, I feel that we should be a little bit broaden our areas. And what I have put title is research is beyond borders. And I gave this example. I generally, when I use the slideshow, I don't want to show everything to you in a first go, but since slideshow has some issue. So he's a person who got a Nobel Prize in 2010, Dr. Venkat Ramans, all of you know. His popular name is Venki, and he prefers to call him by Venki, not by Dr. Venkatraman or Professor Venkatraman. So he got a Nobel Prize in 2010. And what is his area? When I looked into it, he studied BSc, MSc, PhD, and post-PhD even in physics. Work on the biology. That means he tried to understand how the amino acid biosynthesis or translation happens in the bacteria, that how tRNA recruits a specific amino acids and so on. That's what he studied, biology. But probably Nobel committee did not realize, or maybe something was wrong with them, that they gave a Nobel Prize in chemistry. So you tell me what is the subject borders that you would like to put, and this is not the one story. In the next slide, I have shown from 2008 to 2021, all these red are completely biology areas where the Nobel Prizes were given. So don't restrict yourself whether I am a biochemist or chemist or botanist or zoologist or so. For example, I did my MSc PhD in biochemistry, but if I don't identify the plant, I cannot work on plants. If I don't know what insect, it's very difficult. I need to dissect the insect and find out which is the digestive system. So I need to be like a zoologist as well. Then if I want to identify a molecule, I need to be chemist. If you want to clone the gene, I need to be molecular biologist as well. So now these subject borders are okay till you are graduate, BSc level. But after that, I think you should broaden your scope and try to be more up like understanding and discussing the bigger things. So this is, again, I don't want to say the slides. All of you know medicinal plants, but I feel all plants are medicinal plants. Because we know something about haldi or awla or tulsi or aloe vera or even neem, that's what we know. But I feel that in all these plants, there is something which is very important. What is important is systematically identify and characterize. Probably Hanumanta was not a taxonomist. He could not identify which plant he has to bring. So that's why he brought a Parvat, and then the Vaidya has to identify and give this to Laxmikant. So what the message from this slide is, this traditional knowledge of medicinal plant was very much there. And not only in our part, even in China, there are like records, something like 2500 BC, these people were using the plants for treating several diseases. And morning you heard about this Artemisia annua. So this is in Maharashtra or even in India. So in Maharashtra, we on Diwali days, we take a Abhyanga Snan. And for that, we use this plant Davna. So, and even traditional, there's tribal communities, we call them tribal communities, but they are like more advanced than us probably. We call them Adani, but I think probably we are more Adani than them. So they use this plant even for treating fever and so on. So all these plants, I mean, this information is very, very important. So this traditional knowledge, and if you think about even today's drug, for example, Respirin, one of the very, I mean, extensively used drug coming from our Sarpagandha, Rauwolfia serpentina. Morphine, which is actually used a very highly, I will say, painkiller used in brain surgeries and so on. It is coming from opium poppy. We called it as Afu. I mean, so all of you know. And artemisinin already we have described by somebody else earlier, so I don't want to go. So these are the, I will say, traditionally known. But what I thought, and since this is a bioactive molecules conference, so I wanted to share, this is the information available in the websites as well as even you can ask Dr. Google also, you will be able to find out. So I just put just few examples. So there are more than 110 molecules coming from the plant and are used as a drugs today. And I'm giving this example, for example, metformin, which is used for diabetics, yeah, is coming from the plant. Taxol, which is used as an anti-cancer molecule, coming from Taxus brachyta, which is again another plant. Artemisinin, we talked about. Aspirin, all of you know that we take aspirin. Dioxin, or even now there are some formulations coming with curcumin and piperine, which is coming from haldi and our black pepper, which is called, what is it called? What is a black pepper? I forgot the name. Mire, kale mire. So all this is actually, I mean, most active and most interesting compound that one should study. And this is only one slide I'm showing for the, especially for MSc and PhD student. These are the actually two PhD students or just finished, I mean, not even finished their PhD. They are BSc and MSc students. They wrote a very interesting review, which was published again last month, called natural product drug discovery in the artificial intelligence era. So what they did is they tried to collect the information. I mean, I will say what is today happening with artificial intelligence or machine learning. And if you have a traditional knowledge and if you are good in biochemistry, this is going to be a tomorrow's time. So you need to, because information generation is not a big problem. Making sense of that information is going to be a biggest challenge. And for that, human interference is required. Whatever AI is there. So all of you know, if you try a Google and in Gmail, and if you write a mail, if some name is wrong, it will come as a wrong. So it doesn't have that correction. So that's why human interference is very, very important. So what I'm trying to say that this is one of the very nice journal, and they have wrote their review during their BSc and MSc. So you should think about it. You should really think about, I mean, at least understanding such kind of information which is popping up. So during the COVID, I was discussing some of with senior colleague that since in the March, lockdown was there and most of the students were sitting at home. So I said, since we work on plant specialized metabolites, we have library, structural library of more than 20,000 compounds. And fortunately, the structures of 3 important proteases from the COVID-19 were published. Structures. One was RDRP, another was MPRO, which is a protease, and third was angiostatin converting enzyme receptor 2, angiostatin receptor 2. So these structures were known. So we thought, since you are sitting at the home and instead of going into the depression, can you select, can you screen these 20,000 molecules against these targets? And they come up with this screening and then screen 7,100 molecules against these COVID targets. And this paper was accepted within the 5 days in American Journal. And today I'm very happy to share with you that this has been cited today in probably 250 times or so. So that's what happens. I mean, and several of the people from the West, they have taken some molecules and they are already in trial. As Indians, we have a lengthy system to get it, but at least we conducted some of the trials for some antivirals and they are showing some promising data. But unfortunately, it takes a lot of time in our country to handle such situations. So now first question comes in my mind, or it should come into everybody's mind, that why there is so much molecules and then so many structural variants in the plant. Because if you see in the yeast, there are only 550 compounds. But if you take any tissue of the plant, there will be at least 20,000 compounds. Okay, and I already answered you, because they have to have more flexibility to cope up with the nature. And there are more than 200,000 such compounds are there. So you can imagine that how much big diversity that exists. And till this, like last 2 years, we were knowing that there are 3 types of specialized metabolites or secondary metabolites, 3 classes. But now there is another class which is coming up, and these are the peptides. So for example, terpenoids, alkaloids, phenylpeptides, a new class is added as a, actually I will say peptides in the secondary molecule. So this is the core question that we are asking in the lab. So how and why this diversity form? Of course, I call them as a nature's chemistry protocol, because we, as a chemist, we develop a lot of protocols, but we need to understand nature's chemistry protocol, that how they synthesize this compound. So of course, there are several biosynthetic pathways that are working. Again, why these biosynthetic pathways are developed and so on, I think you can look. But what is most interesting for me is that these pathways are very, very specific. Some of the pathways are very specific to plastid or chloroplast. Some of the pathways are very, very specific to cytosol. And then there are also I will say the membrane barriers. What can be moved across the membrane and what cannot be moved across the membrane is also very much decided by this cell. So you think about like just terpene, there are monoterpenes, there are sesquiterpenes, there are actually diterpenes, triterpenes, and tetraterpenes, and all these are synthesized in a very systematic way from a single molecule called IPP or DMPP. So how this molecular diversity is created by the plants and how this machinery is so precisely working in the plants, and that's what we are trying to understand in our group. And of course, there are several groups, and there are even much more complicated pathways, which I will talk a little bit for more detail. For example, you don't have to remember name, but there is something molecule called alpha-tomatin or alpha-solanin, which is produced in tomato and potato respectively. It has more than, I will say, 20 different genes or enzymes, which are systematically producing part of it We call it part 1, part 2, part 3, and part 4. And then finally, you get these molecules very nicely synthesized in the plant. Also, this phenyl pathway, where we also contributed something that how these, especially 4CLs, which are here, how they divert the flux from the lignin biosynthesis to the specialized metabolites. And we have characterized several of these molecules from Ossifum species in detail. But what is the link between these primary metabolites and secondary metabolites? So we somehow, at some time, we name them, they are secondary and somebody is primary. We are very famous to do that categorization. But when we look, there is a nice crosstalk between this primary and secondary metabolite. There's a nice communication. What has to be synthesized, what has to be not synthesized, and most of the substrates for specialized metabolites comes from the actually primary metabolites. And now we'll ask, since this is a slide I put only for students, that when we want to analyze the metabolites, we typically use some kind of these methods like thin layer chromatography or gas chromatography or HPLC or LCMS. I'm not going to go into detail of it, or something. If you want to understand volatile, that means what is coming out of the plant, we called it as a headspace measurement, or we called it as a actually solid phase micro extraction that we can analyze and we can see. And then all these techniques has advantage and disadvantage. But what I wanted to say, use particularly this one, that there is no any solvent, universal solvents that can identify or extract all metabolites. That's the biggest challenge. And none of this technique also can identify all the metabolites. So that's the biggest challenge. And in recent times, this mass spectrometry has actually made some revolutionize to really understand this. Again, as soon as you talk about some high-end techniques, mass spectrometry, we always get scared because this machine is purchased for several crores and so on. But please understand, if you know how to make bhakri, or you know how to even sew something on pali marayche jar mahit asel, so all these techniques are much more simpler than that. Only thing, our hesitation should go on. So all the mass spectrometry techniques are actually based on a simple principle, what I am showing here. She is doing mass spectrometry. Oh, she is separating these grains even based on the masses. And in mass spectrometer, we use something very fantastic term we call collision energy. That means we break the molecule and then we try to analyze. She knows very well how the wind is moving, and accordingly she moves. So observe things around you. I think they will give you better ideas. And I'm happy to share with you that 4 or 5 Nobel Prize in last 20 years went to discovery of mass spectrometry, some techniques in mass spectrometry. So think about it. Like, I mean, I give another example. So in Naneshwara, this 25th OV is मनोनी जाने तेने गुरु भजिजे तेने कृतकार्य हुईजे जैसे मूळ सिंचने सहजे शाखा पल्लव संतोषती. So means what? I mean, in our country or in our region, we know I by heart Naneshwari from first to last, and somebody will say, no, I'm even by heart from last to first. But we did not understand what he says. He says, what is the say, madam? Shaka pallava santoshiti. Jaise mul sinchane sahaje. That means if you want to prosperous the plant, if you want to plant to grow and give a nice yield, you have to water the plant at the root. This is an information. Okay, now all these technologies which come from Israel, whether it is a drip irrigation or it is sprinkler, what it says? It says that the water should go to the root instead of wasting everything. Okay, we kept saying that we know Naneshwari this way, but we never understood what the message coming from this. So this is what I was trying to say, that you please look around you, and this is actually more of an information bank where you can cash upon, and we can really develop a new techniques. And so if you do the headspace measure analysis, you will come up with such kind of a chromatograms, and then there are ways to find out what that peak means, and so on and so forth. So beyond that, like for example, if you use a simple such technique, so like this is a strawberry that we eat. So we found these are the major compounds which comes as a volatile compounds, linalool and neraldol, which is a monoterpene and sesquiterpene. But in the wild strawberry, which is not domesticated, it doesn't have these both the compound, but it has different kind of monoterpenes and sesquiterpenes. And during the process of domestication, probably we have lost some of these compounds. So not only that, we can use this LCMS techniques, which is called mass fragmentation. You can identify hundreds of molecules, not only identify, but you can quantify, because for example, if you see all these ligands here, these are the, like, for example, potato tissues that I have analyzed when I was in the Weizmann Institute as a C.V. Raman fellow, the tuber flesh, tuber peel, whole tuber, and so on. And you can quantify. That means this compound is very high in these tissues, like for example, in flower parts, whereas it is much lower in the other tissues. So that's what you can even not only identify, but you can even quantify these compounds. So today I'm not going to talk about fruits. I'm not going to talk about even, I will say, serpegin. I will bit focus on osmium and steroid glycoalkaloids that are present in tomato and potato very briefly. So all of you know osmium is very important. Again, no need to tell you. But what is more important is when we looked into the literature, that people have did a lot of studies, take aqueous extract, take water extract, and show it has anti-inflammatory activity. Nobody has really looked into the detail which molecule is important. So our attempt was to look what are these metabolites. So we could see that there are some species which are rich in sesquiterpenes, some rich in phenylpropanoids, and some of them rich in even monoterpenes and other molecules. And according to that, we have distributed that. But more importantly, we, as I said, that we look into these trichomes, and we feel these are like very exciting, that we can identify or we can even characterize several biosynthetic pathway when we study, because this is a very clean tissue. These are like single cells or multiple cells, but they are avoid of these like housekeeping genes, such as photosynthetic genes and so on. So we are focusing on that. But then while studying this, we developed, one of my friend, Mahesh Kulkarni, develop a simple assay based on mass spectrometry to see, because in diabetes, there are two major issues. One is your glucose level goes up, and the another is glycation. So all of you know that as soon as extra glucose is there, it is very notorious. It doesn't sit like that. It binds to something else. And that's why when somebody has a diabetes, they say that you monitor glucose as well as HbA1c, which is, it has to have value between 5.6 to 5.7 or something like that. If it goes beyond, means the glycation, if it goes high, it is a problematic, and it creates a lot of retinopathy, nephropathy, cardiopathy, and so on and so forth. So he developed a simple assay based on mass spectrometry. If in any extract, if you have a molecule which can inhibit the glycation, we can identify. And by using this simple assay, we screened several osmium species, and we found in osmium gratissimum leaf extract or inflorescence extract shows very high glycation inhibition. And this is probably, we thought it is a very interesting molecule. And when we looked into the osmium gratissimum, we found this eugenol is very high. And it also shows, I will say, concentration dependent glycation inhibition. So we looked into further, and we did some in silico studies. I think you don't need to know more detail about it. But what is more interesting is, once we have some evidence, that in vitro evidence, we use this molecule to look what happens in the diabetic animals. So we introduced the diabetes in the animal, and then we injected this molecule called eugenol, and we found, very surprisingly, apart from HbA1c, glucose level became normal. And this was very, very surprising for us. And then when we looked into the detail, what we could see, that this molecule is inhibiting even glycosidase. So that means it is helping to have a slow release of a glucose, because as soon as we eat something, starch, we don't use that starch as is. We have to digest with an enzyme called alpha amylase, glucosidase, and so on. This molecule actually, it is not actually I will say, completely block the digestion. It allows glucose to have a slow release. And most of our local land races of sorghum and even I will say bajra, rale, I mean, these all millets have such ability. But now we have shifted to wheat or rice, and that's easy for digestion. But we did not know that easy for digestion could be a problematic. That's why India is, I will say, what they called hub of diabetics. Yeah, because we shifted our local habits. When I was a kid, I used to get chapati only during festival or somebody guest comes. Oh sir, bhat kadi milaycha, kuni jar aala ta pauna. Now, I mean, you guys cannot live without chapati. I mean, if your parents say that you eat bhakri, they say no, it's not good. It's not prestigious. पाहुणे आले तर आम्ही साखराचा चहा करायचो, नाही तर गुळाचा करायचो. आता साखरेपेक्षा गुळ महाग झालाय. In Maharashtra, it may be true everywhere. So now wheat is cheaper than wheat. Wheat is cheaper than sorghum and pearl millet. So please understand our traditional habits. Don't really get carried away with pizza and burgers. They are okay sometimes. I also eat sometimes, but not regular. So we identified this molecule is doing wonders. And we did a lot of, in silico studies. But what I wanted to show you is that when we feed this, when we injected this molecule, we see that pancreas, kidney, liver, and everything is showing a lot of improvement as compared to the respective controls. Yes. See you. Yeah, I understand. Yeah, that's what tight schedule happens with the scientist. If you are a good scientist, you have to keep traveling a lot. Okay, so what? Let us go back to the story in a nutshell. Yeah. So what we found out, this eugenol is a molecule which has a dual role in diabetic management. And how it is dual role? Because it inhibits the alpha glucosidase and have a slow release of a glucose. On the same time, this also competes with glycation with the glucose, and it binds to the same position where glucose would like to bind. And that's how it is a dual role. So now when we looked into this molecule where this is very high amount, and to surprise, I will tell you, it is not a very high level present in our osmum. It is high level present in cloves, launga made. Now everybody understand Marathi here. Huh? So the people who did not understand Marathi, they were left already. Looks like. Okay, so that molecule was highest in cloves, and you know, from ages we are eating something made up of clove after a meal. So it's a logic. I mean, there is a logic behind it. It is not, we don't want to say that it's non-scientific. It's very much scientific. Unfortunately, that time, scientists don't have the mass spectrometer. So that's why they did not find out, or they don't have this assay developed on mass spectrometry to check. So we had an opportunity to do that, and we should do it. So this is one story. Coming back to the another story again, bioactive molecules. So all of you know that these steroidal glycoalkaloids, which are present in the solanaceous plants such as potato, tomato, eggplants, and so on, they are found in some plants of Liliaceae as well. But more interestingly, these compounds were found something like 1820. That means 200 years back, these compounds were actually identified by a French group. And all of you know, traditionally, eating such potatoes is not good. And there is a law in developed countries that if that molecule is X level, these varieties are banned. But in our country, we say that we have so many people, somebody dies also, we read the news next day, we feel very bad, and the next day we forget. That's what it happens. So we thought looking into these molecules in further detail. And when we looked at the literature, we found that this molecule is produced from cholesterol. And it was much more difficult to really look what is known about this molecule. So we know that these molecules are present in potato and tomato, alpha-tomatin or solanine, but we also know that probably cholesterol is the substrate. But all these dotted lines means that we do not know which are the specific enzymes and specific substrates or intermediates which are formed in a production. So in one first part, this was done by Prashant Sonone, that he discovered the pathway of cholesterol in the tomato plants. Don't tell me the plant don't have cholesterol. Plant do have cholesterol. Plant do have cholesterol. Only thing, it is not in the gram level, it is in milligram level. And this was a paper we published in Nature Plants some time back. By the way, I can tell you that in humans, the cholesterol pathway was discovered in 1964, and somebody got Nobel Prize. And same thing was for even other fungi, but unfortunately plant, there was no pathway was known till this point. So we looked into it, and we used all omics. I'm not going to go in detail, transcriptomics, proteomics, and metabolomics to understand how this pathway is functional. And we come up with the 10 genes which are actually involved using various techniques, which I will describe a little bit to you later. But these are the different genes that we identified, which was involved in the cholesterol biosynthesis in tomato, and which similar pathway do exist in the potato as well. So these are the different genes that we characterize. Again, so this is I will say, the pathway that we discovered from these steps to these steps, and what we could see, and that's the beauty of biochemistry as well as chemistry or biology, that these enzymes, what you see in yellow color, they are same enzymes. They can use the substrate, and we can make the intermediates for brassinosteride or sigmasterol pathway as well as cholesterol pathway. But then green color or this pink color, these are the enzymes. For example, if you see, this is oxidase, this is also oxidase, but it doesn't take the substrate for this pathway, and neither SMO4 takes the substrate from this pathway. So half of the pathway is actually managed by both pathway, in the sense like enzymes can do for both pathways, and half of the pathway is very unique, and we called it as a nature's patchwork. So that means nature put one patch here, another patch here, another patch here, so that there is no any, I will say, specific pathways based on their requirement. They can control and they can synthesize these molecules. So when we took this all 10 genes from tomato and put into the Arabidopsis plant, you could see the cholesterol amount is increased. Of course, the plants were not very happy. Their size was reduced. And that is one of the challenge when we think about metabolic pathway engineering. We need to understand metabolic flux. And all of you know, a plant, I mean, in our traditional knowledge also, I used to also sometimes laugh why you have to collect, because if you go to Vaidyas, they always say that you have to collect the plant on Purnima or Amosha or mid of the day, midnight or so. And we were laughing that why this was happening. But now we know that even these metabolites are produced by the plant in a circadian rhythm. And a classical example I will give you is Ratrani, for example, starts giving a smell during 5-6 o'clock. No? Why? Because we go from office, so we should feel better. Is it like that? No. Probably they may be attracting the nocturnal insects. So that means the metabolites are there, but they are actually synthesized, or they are modified in such a way that they are actually produced in a specific rhythm. And that is what, again, I will say, beauty of nature. That so even the cholesterol is produced in even Arabidopsis, it's not in such a high amount. So if you overexpress the gene, plants were not so happy. So actually, I showed you briefly that how cholesterol is synthesized was now we could understand, but how this cholesterol is converted into this solanine in potato and alpha tomatin in tomato, I will describe a little bit more detail, and then I will end my talk. So again, we use this mass spectrometry and analyze 24 tissues of tomato, starting from, like, for example, peel of immature green fruits, then turning red, then orange, and then red. Peel separately, flesh separately, seed separately, and then different tissues like root, young leaves, mature leaves, even birds, flowers, and actually even pollen grains. And we could see that there are these metabolites. This red means the amount of these metabolites is very high, and blue means they are not detectable or less. So this is called heat map, basically. So based on their concentration, we can analyze. So to just make you a little bit better to feel instead of seeing this heat map, so we identify there are specific molecules of this glycolaldehyde pathway exist in this kind of fruit. As soon as the fruit is started ripening, these metabolites also get converted into this. And then, for example, in the ripening tomato, these are the like metabolites which are seen. So if you are a biochemist or if you are a biologist, you will understand how this molecule is modified to become like this. So you know that which enzymes, either it is hydrolysation, glycosylation, acylation. So you understand which enzymes are involved. And then finally, this is produced. So how we selected tomato? Because if you eat these tomatoes, probably they are not very good taste, or they have maybe bitter taste. Although in our region, there were some tomatoes we used to use as a green tomato for making vegetables. But probably these varieties were very, very different. So we discovered this metabolite pathways, and we also even went to even further in the tissue where they are synthesized. Again, using a very high-end techniques, sometimes you have to use it. But more importantly, we could identify even two genes. This is called game 1 and game 4, this is glycoalkaloid metabolism gene 1 and glycoalkaloid metabolism gene 4. And we have discovered now more than 30 genes, but we still say game is not over because there may be more games that we are trying to discover. So what is again seen here is this is the metabolite data I showed you in another earlier slide. And you see these genes are co-expressed. So wherever is this red is there, the gene expression by real-time PCR also shows that these genes are very high in this tissue. So that is how you can discover the pathway. And we use this technique further to characterize this is to really identify the potential genes. And this is called co-expression or network analysis. I think, where is a Kamble? So you should really adopt this. This is a very nice technique. So if you bet one gene and use the transcriptomic data available, you can find out which genes are expressed with that. And it's very simple, by the way. So if suppose Kamble is there or Gajanan Zori is there, who will be around it? Sometimes easy to identify, no? Because his friends will be around it. His friends will be different. So that's why this program, the algorithms are developed in such a way that you can understand which genes are co-expressed with that. And this actually can easy from thousands of genes, we could come up with a dozen of genes which probably are involved in this biosynthetic pathway. And very interestingly, when we looked into the chromosome, so out of 10 genes which we identified, 6 genes are actually on chromosome 7 and 2 genes are on chromosome 12. So that means genes are clustered. If you remember your microbiology, we have something called polycystic mRNA. So something like trend also seen, like for example, the genes which are involved in a specific biosynthetic pathway are clustered. They are coming together. And this is also a very new, I will say, knowledge that is coming up. And that's how we discovered this biosynthetic pathway. From cholesterol, there are at least 10 different genes and enzymes which make tomatin. So now how to really understand, how to really put, like for example, if you see, I have put here game 7 and question mark. Here I have put very clearly game 8. So how you do this? How you study? Because we spent considerable time to understand this pathway, but we realize it is better to ask this question to the plant instead of we are answering it. And that's what happens a lot of time when we ask this question to nature, nature will give you answer. So what we did is when we overexpress this gene, we could see that this is really pathway is active. And if you silence, for example, game 8, what will happen? This enzyme conversion will not happen. That means this 22 hydroxycholesterol will not convert into 22-26 hydroxycholesterol. When you do mass spec analysis, you will be able to identify this compound is actually not produced and this compound is accumulated very fast. So we use these gene silencing, gene overexpression approaches to understand and to put forth this pathway, which is completely new to the science. And this paper we published in Science a couple of years back. So when we looked, for example, just I will share the story with game 4. So when we silence the game 4, we could see that some of the plants did not like, sorry, this is game 1 silence plants. So plant did not like, but when you silence the game 4, we could see nicely the plants are actually happy and fruits are also very happy. But what was more interesting is the pathway which is producing solanine in potato and tomatin in tomato, they are actually reduced dramatically. That means by just silencing one gene, whole of the pathway is blocked. And how this is happening, we could see that whole of these metabolites which are actually related to steroidal glycoalkaloid are actually very less in the silence plant where we have silenced the game 4, whereas the other metabolites are popping up. And this is what you could see the mirror image that we identified this steroidal saponins, which are actually increased in the game 4 silence plant, whereas this solanine and chacosine are reduced. So that means when you block this gene, the whole of this pathway is inactive, and the substrate which is actually put to another pathway which is called steroidal saponins, and this is how it actually changed the metabolic flux from one pathway to another pathway. And this was actually probably very interesting. So for example, if you see the absolute levels of steroidal glycoalkaloids in this game 4 silence plant, almost they are not there. That means we can eat these potatoes, even tomatoes, without having any anti-nutritional activity. So when we submitted this paper to Science, they published it after a year with some revision, but they wrote about our paper from nasty to tasty. That means we are actually reducing these toxic compounds to almost zero level by just silencing one gene. When we started this work, it was completely as per the definition of Indian scientists, basic research. When we published this, this patent was purchased by a Chinese company for $6 million. So that's the beauty. So you should not really think about what is the basic and what is applied. Science is science, and if you need to think how you apply. So if you start doing something like jugaad and startups, that's not going to work. You have to really invest science and learn science, and then you can think about how to put together, how to put things together, and you can develop your own company, but not based on the very superficial information, because you will not, I will say, success if you your product is based on the jugaad. Okay, so with this, I thought I will stop because I think I have taken more time. What is happening? Yeah, just a minute. Oh yeah, yes, please. Yeah, so with this, I thought I will, these are the people who were actually involved in the research. I don't want to take name, but I wanted to take the name of the Dr. Vidya Gupta, Tulsiram, Mahesh Kulkarni, Asaf Aroni from Weizmann Institute, Avinash Kamble and Abhijit Kulkarni, Tamane and Kotkar from Savitribai Phule University, Martin Jongsma from Netherlands, and my friend Sachin from Biosphere, and several of these students who were actually contributed to this project. And this is what we always believe on, the joint family. So it's not my group, it's our group. So Dr. Ranjekar, who joined NCL in 1975, he was the person who started isolating plant DNA from the plant. So in the first time in the country. So he started this group coming from Canada in 1975 to NCL as a scientist. Then he superannuated or retired. Then Dr. Vidya Gupta continued this. She also retired. Now myself, Dr. Narendra Kadu, Dr. Kirti Kumar Kondhare and Rakesh Joshi, and now we have one more lady scientist, Archana. So we five manage this group, and these are our actually work horses. These are the students. They make significant difference in the science, scientific understanding, and in the group. So we are actually probably more of a scientific managers. These all students actually contribute to the real science that what we do. With this, I would like to say thank you for your kind attention, and I will be happy to answer any questions. You can ask question in, I mean, since Professor Kadam is saying only one question, doesn't matter. I'm available here during tea or any time we can keep free. But if you want to ask the question in, at least I understand 3 languages, Marathi, Hindi and English. So any language is okay for me. Hindi, I'm not so good, but.
So thank you, sir. Thank you very much for the excellent talk. And because we were following it up. So it was exhaustive data you have presented, because I know it is very hard to present what you are doing in 40 or 45 minutes, because you have spent almost 20 years in NCL, and then doing continuing the same area, and then also exploring a lot of new themes of that particular aspect, right from defense, insect defense mechanism to metabolic engineering. And it was really very informative, very nice lecture, especially for our students. So students might have understood what are the future opportunities for them in this particular field. So thank you, sir. Thank you very much for your presentation. So any question from you guys? One question, and you'll get something as a treat if you are asking a question. Yeah.
I agree with you. Yes, if their plants are niche based, then it is difficult. But what I can tell you, I have not shown you, but if you read my abstract, I wrote something about steropyzine. So steropyzine is an endemic plant species. We have 32 species in Western Ghats, but several of them are endemic. So what we did is we propagated through tissue culture, and we have put them back. So it is difficult to grow in Pune. I agree. Yes. It's not all plants you can grow in the. Just not the environmental factor. There is Professor Lal microgram specific plant has their own microgram to grow. Yes. So how are you getting that? Yeah. So I forgot to. So thank you. You said what we did is when we got this 110 plants, so we went to respective places and we brought a 5 kilo soil from that region and we put just near that plant. So that idea was to have that microorganisms there. But we were lucky, we got microorganisms which we cannot see with naked eyes. But we saw there are several small plants like grasses which are native of that region. So we got this. Our idea was to go 110 plants. But when we saw it, there were like 250 plants, which were of native. So along with that soil, thinking of microbes, we got, but then even the small herbs like grasses particularly and even small herb plant. So it is possible. To mimicking it's possible.
So thank you, sir. And I hope if there is no question, we will have a discussion with you after yeah, question. No, if we say that all plants contain secondary metabolites which are acting as beneficial components for resisting human disease.
So, there, I mean, the question is when we say that all plants contain secondary metabolites which are acting beneficial components for resisting human disease. Yes, I mean, the broad answer is yes, but of course, human disease is more artificial because of, number of things, like for example, COVID, although we call it as a natural pandemic also, it came because of something, some different experiment that we did. So yes, why we are actually healthy? Because why our immune system is active, and in the recent times, a lot of things are talked about immunotherapy or immune boosters and so on. So most of them are coming from the plants. So in my opinion, yes, plant will have molecules which actually keeps our defense system on, not only by just food and nutrition, but also the immune boosters are there. So I think diseases will not come when you are healthy. So that is the role of the plant. So I don't think this ecosystem will exist if you remove plants from it. So plant do have an important role.
Thank you, sir. So you might have realized why we have selected this theme, right? Advances in bioactive molecules. So you have heard about the significance of these bioactive molecules are playing, not just for humans, but even for all the living organisms, especially plants. So thank you, sir. Thank you very much for enlightening us about this plant bioactive molecule. And yes, I knew that today I came to know that tomato has 20 tissues. So thank you for that.
Thank you very much, sir. Thank you for your wonderful and excellent talk. Now, moving towards the next session, the next lecture is Dr. A.B. Ade, sir, who's joining with us virtually. I would like to welcome Dr. A.B. Ade, sir, from Department of Botany, Savitribai Phule, Pune University, Pune. Sir, are you there?
Yeah.
Ade, sir. Am I audible, sir?
Yeah, you are audible.
Yes. Now, I would like to request Dr. T.A. Kadam, sir, to please introduce our next speaker, Dr. A.B. Ade, sir. Sir, please.
Welcome, Ade, sir. Actually, we are late and we have to complete validity programs before 5:30. Ade, sir completed MSc from Amravati University and joined as an assistant professor in Aurangabad University, Dr. Babasaheb Ambedkar Maratha University, Aurangabad. And at the age of 35, he joined as a professor in Pune University, so youngest professor. So his area of interest is bioremediations and plant microbe interactions. More than 10 students awarded under his guidance for PhD. Publication more than 50. He has completed many projects. So I will not take more time in introducing the Professor Ade sir. He's well known. So I request Ade sir, please start your presentation. Ade sir.
Yes, sir. Thank you very much. Can I share my slides?
Yes, yes, yes. You can share.
Okay. Is it visible, sir?
Yes, sir.
Okay. So thank you, Kadam sir, the organizer, for the introduction. My today's topic is plastic degradation and related aspects. So plastic will go through the slides. These are self-explanatory. So very first of all, let us understand what is plastic. So plastic is the synthetic or semi-synthetic organic solids. So synthetic means man-made, semi-synthetic, partially man-made, partially natural. Or all of you may be knowing that these are the petrochemicals derived substances, and these are having high molecular weight, and they are either in the pure form, means homopolymers, or there may be some additive uh, compounds in that, so that forms the heteropolymers. And because of this, they are known as partially natural or partially man-made. Okay. So basically polymers related to this plastic, if we try to classify, then we can make two categories: the thermosets and thermoplastics. So thermosets are the compounds which cannot be melted, and thermoplastics, these can be melted. So the examples are given. The thermosets are having phenolics, epoxides. These can't be melted, while melting plastics are the thermoplastics, and they are further categorized into two: semi-crystalline and amorphous. So semi-crystalline, they are having specific structures, and amorphous, they are just powder. So such type of examples, that is polyethylene, terephthalate, nylon, etc. This can become under semi-crystalline or structured type of polymer. And amorphous, these are polystyrene or polycarbonate. This can be make available as a powder. So after this, let us understand what thermoplastics are. So these thermoplastics are said to be having 20,000 to 5 lakh atomic mass unit, and it do not undergo chemical change in their composition, and they are very sensitive to heat. So they can be heated and converted into molded and remolded again and again. So examples of this kind are the polyethylene, polypropylene, polystyrene, polyvinyl chloride, and polytetrafluoroethylene. Then thermosetting plastic, if we take as an example, then this old generation telephone or this screwdriver, specifically this portion of the screwdriver, that is having a thermosetting plastic, which are quite rigid, and they are not melted, and that's why they are said to be hardened plastic. So these have different composition, these have different components are added in that. So one good example of this thermosetting polymer is this rubber tire. So this rubber tire is having specific polyisoprene units, and this polyisoprene, they are interlinked by means of this sulfur. So whenever the sulfur is added, the vulcanization is said to be done, and due to the addition of this sulfur, the flexibility of this is turned into rigidity, and because of this strength that particular polymer received, and hence it can tolerate the heavy load on the trucks. We can say that if it is supported with the air by means of tube, then about 15% contribution is there from the rubber tire to bear the load, and 85% contribution of the air which is contained in that particular tube. So such a strength this particular polymer gets, and that is because of the addition of the sulfur, and the entire process is known as vulcanization. So such type of polymers can't be melted totally. So these are known as thermoset type of polymers. Other classifications are also possible of the plastic, depending on their different nature. So for example, elastomers are there, structural plastics are there, electrically conductive plastics are there, and finally the biodegradable plastic. So elastomers are the very flexible tubes, and they are having viscoelasticity, and they are having a quite extendability or long elongation of that particular material. So very good example is the elastic polymer, that is rubber. Then next, the elastomers, if we try to classify further, then thermoplastic elastomer and thermoset elastomer. So quite very soft elastomers are there. They are having the polymeric chains of this kind, and they are not connected with each other. So these chains are straight chains, and they are having the sliding mechanism for the stretching. However, in this case, thermoset elastomer, the cross linking is there in between the chain, and because of this cross linking, the rigidity is occur, and hence it is difficult to go for the sliding mechanism or extension. So these are two basic types of elastomer: thermoset and thermoplastic elastomer. Then structural plastic, all of us know. So here the monuments or the toys or different settings in the gardens, in play children area, this is made up of plastic, because it is sturdy one, and that is not affected at all by any of this sunlight or raining or winter or cooling. That's why in open area this is to be kept in very sturdy manner. So in this way, such plastics, they are available for making these monuments and these things. These are known as structural plastic. Then electrically conductive plastic. So here the plastic is connected with some additional compounds to have the partial conductivity of the electricity, and because of that, this is known as electrically conductive plastic. Otherwise, plastic is insulator for the electricity, but due to addition of certain chemicals, that become you can say conductive for the electricity. Then biodegradable plastic, that is there. So biodegradable plastic is actually the contradictory term. On one side, we say that the plastic is not degradable, and on another side, there is a biodegradable plastic. So what is there behind this? Biodegradable plastic is the amalgamation of the polymer of this derived from the petrochemical, means these are derived from the fossil fuel, and in addition to that, we are adding certain organic material in that, or bio derived material in that. For example, starch, if it is mixed with the plastic, then that becomes the biodegradable plastic. So such type of biodegradable plastic nowadays the manufacturers are going to produce, and this is also available in the market also. Then based on different properties of the physical properties of the plastic, these are classified according to this. First, the density. If the density is high and low, then HDPE and low density polyethylene, that may be the example. The tensile strength, that will be the criteria for classifying the plastic material. Then glass transition temperature is the criteria for the classification of this plastic polymer. Then resistance to various chemical products, that can also be the property, physical property by which the strength of the plastic that can be determined. So one by one, let us see. Density dependent plastics are having examples of HDPE or LDPE, high density and low density polyethylene. So by observing this picture, we can understand very easily. Then tensile strength of the polymers, if we try to see, then this is a list of polymers and possible tensile strength. So where the uh, plastic material comes, that we can observe here, and according to that, the specific tensile strength is provided with that. And the percent elongation and tensile modulus, these values are also given. So elongation, if we try to see, then highest elongation is seen in polyethylene, specifically high density polyethylene. So these are certain physical properties. Then glass transition temperature. So this is a temperature in which the plastic is converted into a amorphous material, which is a rubber-like substance. So any of these polymer can be converted into rubber-like substance at this particular temperature, and that temperature is known as glass transition temperature. So according to that temperature, the polymer, one can have the specificity of that particular polymer. Then coming to the resistance property. So resistance to different chemicals, we can see even nowadays the medicines are appeared in the plastic bottles. So these are not you can say reactive with the medicines or strong acids. Even you can put the aqua regia or hydrochloric acid, nitric acid, all these. If these are put in plastic bottles, then these are kept safe, and that's why nowadays instead of glass bottle, the plastic bottles are preferred by many of the manufacturers. So such type of resistance is observed by the plastic material. So that is known as plastic resistant to chemicals. So because of this particular properties of the plastic or polymers, the consumption of the plastic is increased, especially in India. If we see, then packaging is a sector which is having 35%, then followed by it the building and construction, then afterward the uh, transport, electronics, agriculture, and some other minor miscellaneous categories are there. So these are formed 19%. So this type of consumption, if it is there, then what will be the prediction? What are the predictions made, or what were the observation made in different years? In 1996, this was the situation of the plastic. Now it has been increased and it is further increased. So how the consumption of the plastic is being done in India per year, that has been shown here in this particular chart. So one basic polymer is the polythene. We are concentrating on that. So polythene is having a common name as polythene, then scientific name is polyethylene, abbreviation is PE. IUPAC name is polyethylene or polymethylene, and this is the chemical formula. So this particular polythene has been focused in our lab because we are concentrating on the management of the waste material, solid waste material. And out of the total plastic polymers, polythene is forming majority of the component of the pollutant, that is 64% waste has been derived from the polythene material. So by considering this polythene, if we try to see, then this is coming under the thermoplastic polymer. category, which is consisting of hydrocarbon chains, the melting points as per the high density and low density polyethylene. These are as shown 120 to 130 degrees Celsius, while 105 to 115 degrees Celsius. And basically it is hydrophobic in nature. That's why this can be used for the lamination, not only for the our certificate, degree certificate, etc. But nowadays the vegetables are also being laminated because of the polythene. So that's about the physical properties of the polythene. Then coming to the chemical properties of the polythene. So resistant to chemical, as we have seen, the strong acids and bases, gentle oxidants and reducing agents, they are also uh, do not causing any harm to the plastic. Then burning property of the polythene, if we show, the burning of the polythene is taken place very slowly with a blue flame, and this flame is having yellow tip in the center, and it gives off a odor of the paraffin. Then solubility is there. Solubility is very critical. So usually it is not soluble at all, but at elevated temperature, the aromatic hydrocarbons such as toluene or xylene or chlorinated solvent, the examples are trichloroethane or trichlorobenzene. This can be utilized for the solubilization of this polythene. So otherwise it is not soluble at all. And because of this particular property, the durability of this polythene is increased too much, and because of this increased durability the usage has been increased, and because of the increase in the usage the pollution of by polyethylene that has been increased too much. Then coming to this pictures, two pictures, one is on the ground and another is in the water. So both of these are having the accumulation of polythene waste or plastic waste, and because of this accumulation, and degradation rate is very slow, accumulation rate is too much high, and that imbalance has been resulted into the pollution. So according to the survey, it has been estimated that 10,000 tons per day of plastic waste is generated in India. So we can think about what we are going to produce and what will be its consequences. Then coming to the waste category, if we try to see, then thermoplastics are constituting 80% of the waste of the plastic material. Then thermoset plastic constitutes 20% approximately the waste, and this forms the combination of this total plastic waste. Then if plastic is not disposed properly, or if there is no any awareness for the proper recycling of the plastic, or management is not observed up to the mark, then this type of miserable condition we can face. So this particular you can say picture is showing, this is the site, tourist site of interest, and people they are consuming the things and throwing the polythene here along the road, and that's why beauty of this particular spot has been gone. In another picture we can see the clogging of the channels, water channels is there due to accumulation of plastic, and because of that, if this channel is totally blocked after some time, then what will happen? The water will not be moving with this channel, but it will be above this particular bridge, and flood like situation one can face. So here these are the hazards of plastic mismanagement. Then we throw the plastic as we even the garbage bag, we don't know what quality of the garbage bag we are using, but if these are not degradable at all, and if we are tying the food material within that and throwing this garbage bag in the site of say dumping, then the food material is there inside. The animals, they don't have any facility to you can say open that carry bag, and that's why along with the carry bag they will be eating the food material. So such type of food material will be rich in polythene or other polymer bags which will be consumed by the animals, and because of that the complications are there in their health. So this is the picture of the dead bird. Here the feathers and other bones and all this, that is showing degradation, but the plastic which is consumed by it, that is not degraded. So it indicates how hazardous it is and how recalcitrant it is. Then coming to the fish, so fishes, they are also consuming the plastic material. Many microplastics are consumed by them, and they can't even differentiate the plastic material from the other substances, because fishes are having unique property to take the water inside, and whatever the necessary material is there, that is to be digested, and remaining material is to be thrown out. But the excretion phenomenon, that do not allow the entry of plastic like material for the excretion purpose, and that's why this get accumulated in the fish's body. And if these are consumed by human being, then plastic material is ingested by us, and that is ultimately causing the cancers, birth defect, immune system suppressions, and developmental problems in children. So one plasticizer is there in the plastic material, that is said to be bisphenol A. So this bisphenol A is said to be the you can say endocrine disruptor, and this can mimic the hormone estrogen. So if a particular material is trying to mimic the hormone, then entire you can say endocrine system will be collapsed, and as endocrine system is collapsed, then regulation of the human body will also be defected, and ultimately we have to face certain problems related with our endocrine system. So such bisphenol A is added in the plastic in order to create the more flexibility or more you can say malleability of that particular plastic material. So for this particular purpose, bisphenol A is added, but on the other side, this is having the hazardous effect, as it is disrupting endocrine system. So disrupting endocrine system is losing the control of brain over rest of the organs, and if this is done, then naturally our body has to face the problem. Then phthalate is one more compound which is mixed with the plastic, and that is going to produce the thinner type of films of plastic. But at the same time, phthalates are causing the birth defect. They are causing the cancerous properties, breast cancer, and so many things in human being. Certain containers of the cosmetic material, that is consisting of phthalates, because nowadays the soft bottles or soft tubes we are preferring. So for making that kind of container, the phthalate is mixed with the plastic material, and within that container the cosmetic creams or allied substances can be inserted, and these are now available in the market. But what happens if we apply this cosmetic, if a particular nail remover is there, so nail remover, if it is there in the phthalate bottle, then we can say that the nail remover, that acetone or that kind of say chemical, that will easily remove the phthalate, and because of that removal, the mixing of the phthalates with this cosmetic product that may be there, and this product along with the phthalates, if we apply on this our skin, then hazardous condition we can see. Then coming to this bottle of the soft drink, that is also made up of phthalate along with the plastic material. And because of this, the phthalate is ingested in our body, and this is acting as carcinogens, and it is causing the defect in male reproductive system in case of infant. So if it is there, then obviously we are facing problems because of that. So what is the solution if these are the problems so many associated with the plastic waste? Then what is the solution for the plastic waste management and how it is to be tackled? Because without a plastic we can't live. A simple example I can give, that milk bag. Early in the morning we take milk for making tea. So milk bag is available, that is consisting of the plastic, uh, that is there in the plastic. That's why milk we can bring from the shop to our home. If plastic is not there, then it is difficult to bring. Every time we have to carry the boxes or tiffins to collect the milk, and then the collection of the material, milk like material we can bring. So because of this plastic discovery, this you can say uh, process has become quite easier. So such type of things we are essentially required in our daily life. But at the same time, the waste management program we should design, because it is our product. We have decided, we have uh, manufactured it. Human being have make the plastic, and it is synthetic or semi-synthetic, it may be, but ultimately we have designed it, and that's why we have to find the solution for the management of it, because we are the part and parcel of the ecosystem. So we as the component of ecosystem has to regulate the ecosystem, and for that particular purpose the waste management is essential. So plastic waste management strategies, if we try to classify, then landfilling is one, mechanical recycling is there, then biological recycling, then thermochemical recycling, that is also there. So in mechanical recycling, further classification we can see the primary product, the secondary product. So primary product will be the product which are derived from the originally existing product.
हॅलो
Like
सर
that.
सर
Yes, sir.
सर थोडा आवाज क्लिअर येत नाही आणि आवर गेस्ट आर वेटिंग फॉर व्हॅलिडिटी प्रोग्राम्स
Okay, okay. So I will try to make short. So
प्लीज
such type of,
कन्क्लूड
yeah.
प्लीज कन्क्लूड
Yes, yes. So these strategies are there, and according to that strategy, our lab has started certain experiments. So I am going to focus on that directly. So these are the theoretical things. So our lab is focusing on the biodegradation. So for that particular purpose, we are using bacteria and fungi which are soil borne. Some of the bacteria and fungi we have already used, and these are their pictures or the micro photographs. And according to that, a simple experimental setup is there, and these are the products of the corroded plastic or degraded plastic, and these are rest of the you can say observations. I'm not going into detail of that. So these are the degradation products which we recovered from the culture filtrates of that particular fungi, and this is for the analysis of the corrosion of the surface, that is FTIR analysis. And then culture filtrate effect on the seed germination, in which we could find that is harmless for the seeds. And this is the mangrove ecosystem that is having richness in the plastic degrading microbes, which we have isolated and applied on the plastic polythene material, and degradation we can see here in the scanning electron microscopic photograph. So because of that, the remarkable publications we could do, and finally uh, uh, the human being has to manage the plastic waste. Otherwise, after some time, this will be the situation. Entire earth will be under the plasticosphere, just like atmosphere, hydrosphere, and lithosphere. So this is the message from this picture. So I am thankful to the organizer for giving me an opportunity to interact with the audience. Thank you very much.
Thank you, Ade sir. Actually, Giri sir explained human beings are more selfish in nature. Okay, when you compare with the other animals or plants, we are only bothering about ourselves. That is anti-cancer drugs, anti-tumor drugs, antibiotics. We are obtaining from plants as well as microbes. If we are using streptokinase, streptodornase, we say it is a therapeutic purpose. But suppose we are using enzymes for plastic degradations, we will not give the word for the therapeutic. We will not give word as a bioactive molecules. But enzymes which are involved in degradation of the plastic or recalcitrant components are equally important as antibiotics, equally important as a anti-cancer drugs. And apart from that, management of such waste in nature is also very important. Actually, Ade sir, you have presented very nicely, but due to time's constraint, our organizing committee also can't provide more time to him. For next events, we'll manage his time and more exposure will be given to the students. Okay, thank you. So I request Vadagalkar sir, please join the conference.
Hello.
Welcome, sir. And now our next speaker is Dr. Prasad Vadagaonkar, sir. He is joining with us from Amravati University, Amravati. Sir is head of the Department of Biotechnology. And I would like to request Dr. Kadam, sir, to please introduce our next speaker, Dr. Vadagaonkar, sir. Welcome, sir.
Thank you.
Welcome, sir. Only 30 seconds I will time take to introduce Vadagalkar, sir. Actually, nowadays we know NET, SET, GRF examination is now become very easy. But in 1986, government made GRF NET compulsory to the teachers. And nobody was qualifying at that time. So first professor or lecturer, teacher qualified with GRF CSR is Professor Vadagalkar, sir, in 89. And with fellowship, he completed his PhD work. He joined in 95 at Amravati University. He has vast experience of research. Presently, he is professor and head, Department of Biotechnology. Different honors, medals has been awarded to him. More than 10 students, 12 students completed PhD under his guidance. More than 9 projects of DST, DBT, he has completed. Then publication more than 50. He remained active in university administration also. He remained member of management council, then director of UGC Human Resource Development Center, and many other membership in the capacity of the professor and head. He remained membership of the scientific society like Chhattisgarh Botanical Society, Association of Microbiology of India, Indian Science Congress, Society for Basic and Applied Mycology, Biotechnology Research Society of India, Asian Federation of Biotechnologists. Okay, so I will not explain, mention all these things. Okay, I request Vadagalkar, sir, please start your presentation, sir.
Thank you, sir. Am I audible?
Yes.
Hello.
You're audible.
Yeah. How much time I will get, sir? I will just try to finish in that time.
Please, within 30 minutes, sir, please.
Oh, it's more than enough. No problem. Thank you. Sir, I will take a 2 minutes to reset it because it is on a different system. My slides are visible to you all?
Yes, sir.
Yeah, actually I'm on a Linux system and it is giving me a problem, but no problem. If slides are okay, then I will move forward.
Yes, sir. Definitely.
Yeah.
Please go ahead.
Thank you. Thank you very much. Let me first say thanks to Dr. Kadam for adjusting my time. I was supposed to deliver the talk yesterday, but I couldn't get the time. I'm really grateful to him that he has given me an opportunity to interact with you all. Since say last 10-15 years, my lab is working on a bioprocess. I'm basically a biotechnologist with my field of interest is bioprocess, where I try to develop the bioprocess for various systems, including from the microbial plants and animal cells. And the topic of today's is the bioactive, advances in bioactive molecules. And one of the theme which our lab is dedicated to validate the herbal medicines, or you can say the bioactive molecules which are present in the plants. We try to validate their actions. And in this area, most of the projects supported by UGC and DRDO, where we worked on this line only. And actually, if you see, we were very much fascinated with this triangle, which was proposed by Marshalker way back in 2005. You can say that our life started during that time only. And we are very much fascinated with this particular triangle, which he was used to say this is a golden triangle, where we had to integrate modern medicine, traditional knowledge with the help of science and technology. As a biotechnologist, we were playing with the different organisms like microbes, plants, as well as animal cells. We decided that we will keep this as a one of the area for our research also. So we move forward and we started talking about medicinal plants, herbal medicines, and how to integrate this with the modern medicine. We know that that time the term was something like alternative medicines, it not recognized. Medicos never recognized the alternative medicines, but slowly even NIH started talking about the alternative medicine, and they said it is a complementary medicine. And they started saying complementary and alternative medicine. During that time, around 2010, 2015, the CAM was very, you can say, the very popular word, complementary and alternative medicine. But during this whole period, not only in India, but around the globe, people recognize that herbal medicine is also having a great role, not only in the maintenance of the health, but also in the therapy. And the term which has been now recognized as integrated medicine. And even the MBBS, MD, MS started allowing this therapy in the mainstream. And especially if you see this, the in cancer therapy, this was more acceptable because people, there was no alternative, and only the chemotherapy or radiotherapy is there, where they don't have actually the mechanism to differentiate the normal cell with the cancer cell, and all the drugs are cytotoxic. Maximum drugs are cytotoxic. So they started believing in this integrated medicine, but problem is with our system, that is our herbal system, is that still we are not in that position to explain the mechanism for each and every therapy which is prescribed by herbal therapy or Ayurveda or Unani or Chinese medicine. But yes, with the help of modern science, now it is becoming possible. So one of the study which I'm trying to share with you is carried out by one of my students who has completed her work. And I'm proud to say that when she joined the department, when joined the lab, she was having a zero experience in the wet lab. She is a bioinformatician, MSc in bioinformatician, but fascinated with the wet lab research, and she insisted that she want to work. She was also an Inspire Fellow with the fellowship, so there was no way to say her or deny her wish. So I decided to leave the topic to her and develop her the topic itself. And I decided that I will only guide, or I will only ask the question to her, and she will carry out the whole experiments. She will learn the things, we will try to answer. And if it is possible, we will convert that work into the doctoral thesis. So she was, first thing, she was very much fascinated with this particular organism, that is a Gynoderma lucidum, a fungi. And she started working, as I said, that she's a bioinformatician. She started working with the databases and started finding out the which type of molecules are present in this, what type of work has been carried out. And she has come out with the information that more than 150 gynoderic acids and different gynoderic acids are present in this particular fungi. And going further, she has come out with the literature that, and she insisted that people have carried out lot of in vitro work, and they have shown that the ethanolic extract of fruiting bodies as well as the ethanolic extract of um, mycelium has shown anti-proliferative activity against various cancers in vitro. I'm just showing you this slide. Slide is visible, sir? Hello?
Yes, sir.
Yeah, thank you. So almost 8 to 10 different, 15 different types of cancer people have already worked, and they have shown that it is having the anti-proliferative activity. So we started talking, is it possible for us to at least pinpoint the mechanism of anti-proliferation activity, or how the cell is getting inhibited, or can we slow down the growth of cancer cell? So we move forward, and then simply my question was the compounds which we are talking about, the gynoderic acid, and if we think it is going to be an oral route, means this extract is going to be through oral route. Is it satisfying the Lipinski rule? Today it is very easy, and especially with the various software. So she moved to the software like a DipCell, which is our MTech program, MTech software, as well as the mole inspiration. And within no time, she has screened the complete library of ganodic acid, and she has come out with the answer. Yes, this almost all these compounds follow the Lipinski rule. So we move forward and again ask the question to ourselves, is it going to address the problem of ADME, adsorption, distribution, metabolism, excretion, and toxicity? Because when we talk about the drug discovery, and if we see the time required for the drug discovery, and at the end, we see that more than 50% molecules get failed in the last stage of drug discovery because of they don't follow the ADME tox. So again, this was a question, because putting the money, time in the lab, we want to be sure that the molecules or the extract which we are going to test on the cells are really not going to fail because of this type of problems. So again, softwares are available where we can do. So again, this whole library was exposed to ADME software, and we found that most of the compounds qualify for ADME. And then we started thinking about, okay, can we take this extract to the lab? Can we go for the anti-proliferative activity? Since my lab is having the cell culture facility, and we are very well supported by NCCS Pune, and I'm really proud to say that I'm one of those scientists in India, I believe, who can get the cell lines on phone call. Of course, we follow all the procedures, but yes, and see, I got we got a very well support from the NCCS throughout my last 15 years of research. So we got this 4 cell lines from the NCCS. One was a 3T3, that is a mouse fibroblast as a normal cell, untransformed cell. Then Miyapaka, that was a pancreatic cell. Hep G2, that is a liver cancer cells, and B16F0, that is a melanoma cell. And we just screened for the anti-proliferative activity for Gynoderma fruiting body ethanolic extract, as well as the mycelial ethanolic extract. And we found that these extracts inhibit the growth of cells like pancreatic cells, as well as the hepatomas, as well as the melanoma cells. And when we started comparing this, we found that it is having the great selectivity index, especially for the pancreatic cells carcinoma. Around 2.20 is exact, more than double. So it is very specific. It can differentiate the normal cell and target or inhibit the growth of pancreatic cancer cell. I'm saying the inhibiting the pancreatic cells because I in this stage we cannot make a comment that it is a killing the cell. It might be inhibiting the growth of the cells. So moving forward, we were interested to know that this anti-proliferative activity is killing the cells like all cytotoxic drugs do, like necrosis, like acting as a poison for the multiplying cells, or it induces a programmed cell death, that is a apoptosis. And as I said, the student was a from the bioinformatics, so she was always asking the questions. And question was how we can differentiate apoptosis and necrosis in experiments. So simple question answer was that you go and read the books, read the basics of the cell, and basics says that if we compare, if we see the cell membrane, it is a asymmetric cell membrane, which we are already fluidized model we are already studying since the schools. So it is asymmetric model of the cell membrane, and it is very clear that phosphatidyl serine, a type of phospholipid which are always present the inside the lipid bilayer. They are never exposed to the outside in normal cells, but it has been observed that during apoptosis or onset of apoptosis, or you can say that when cell get a signal to die, in a very early stage, there is a flip-flop mechanism, and that too we study in this graduation level as well as in post-graduation cell biology. So there is an exchange of these phospholipids in this lipid bilayer, and phosphatidyl serine get exposed outside. And so there are a lot of tests are available to detect the presence of phosphatidyl serine on the cell membrane, and one of them is the annexin. Annexin is a very specific, it binds with the phosphatidyl serine very specifically, and if we tag the fluorescence to it, we can monitor it. So we started with this, and we just use annexin 5 as well as the PI, which is a dye, propadium iodide, which stain the DNA, which cannot penetrate in the living cell. So when we expose these two chemicals to our cells, which are growing in vitro, and we found in the population very few cells were undergoing apoptosis. You can see very well here that fluorescent green cells, few cells are present, no red cell is present. And if we merge this, we see the green cells, fluorescent cells, which are showing that in population, there are few cells which are going under apoptosis. This is a normal process in all the living system. And then we expose this with the extract, and we found that within 6 hours, large number of cells started showing the fluorescence, which clearly indicates that cell membrane structure has changed. Phosphatidyl serine has been exposed to the outside, and our alexanin is binding to this. And within 24 hours, we can see that red cells started appearing, means red fluorescence is also started appearing, because within 24 hours, the integrity of the cell membrane was started losing. Propadium iodide entered into the cell, reached to the nucleus and stained the nucleus. And we see if we use both the colors, we see the yellow cells, which are clearly indicating the stage that is a late apoptosis. So this experiment has proven that these extracts are not cytotoxic. They are inducing the program cell death. Then we started thinking about, since the number is not increasing and we are saying it is reducing the growth rate, we went forward for the cell analysis, cell cycle analysis. And in normal population means the cells which were not exposed with the extract, we found the distribution around 50% our cells are in G1, G0, G1 phase, 11% cells are in G2 phase, that is here, and around 30%, 38% cells are in synthesis phase. So when we expose this, we found that, I'll come to directly to this, within 24 hours, the cell population moved to the G0, G1 phase. The maximum cells were now present in G0 as well as G1 phase, and very few cells, from 38% has gone down to 12% in the synthesis phase, and G2 phase or dividing cells 9.52. So it is clearly indicating that yes, it is slowing down the growth, it is inducing the apoptosis, it is not lethal or toxic to the cell. But now we started asking question, is it possible to pinpoint the target. So since we are using and all the experiments were done in the pancreatic cancer cell, we started again to the literature search and we started playing with the, you can say the bibliomics in terms of the bioinformaticians terms. And she has come out with the one possible target saying the urokinase plasminogen activator is playing a very important role when these pancreatic cancer cells are becoming resistant to chemotherapy. And we went further move forward and try to study the role of this urokinase plasminogen activator. We found that this is nothing but it is one type of peptides, or you can say the proteases, which play very important role in degrading the ECM, that is extracellular matrix, and allows cells to move forward, or you can say allow cells to migrate. So we move forward and again decided to do a small experiment to check that does our extract inhibit the cell migration or not. See, our all bioinformation in vivo, in silico experiments are guiding us to design our in vitro experiments. So this this literature survey has asked us to find out that our extract is inhibiting the cell migration or not. It is inhibiting the cell growth, it's fine. But is it inhibiting the cell migration also? So we did a scratch assay, which is, you can say, the wound healing assay, opposite of wound healing assay, something like this. So we have used the pancreatic, pancreatic cell. It is written in the prostate. It is a pancreatic cells, and we scratched the cells and treated with this extract. And we found that within 24 hours or 48 hours, we can see the difference. This is a control where we have not used the extract. And here it is cells, very few cells have been migrated to the clean area. So it is inhibiting the migration of the cells. So again, we move forward and we took the help of bioinformatics again, and we tried to find out, is the crystal structure is available in the database of PDB, and it is available, and we found that the structure of urokinase is available in the PDB along with a known inhibitor. So inhibitor which can inhibit this was present in the structure, and that whole structure is available. So we use the molecular docking to confirm this. We use the two software. One is a FlexX, which is a commercial software which is available with us, and Autodoc just to confirm the results. We use the Autodoc, that is open source, and we use the same molecule along with the inhibitor to locate the position where it goes and binds, which goes and binds to the active site of this urokinase, and targeted our molecule to this particular site. And we tried to find out that these whole structures, which has been proposed like a ganoderic acid, around 100, 150 molecules, which are having the almost common structure with the different R groups, can go and bind to this particular site, which is an active site of urokinase. And if it goes and bind to that, it can inhibit this. Because we have seen by experimentation, we have already seen that it is inhibiting the migration. And migration, if we are inhibiting the migration, it means we are inhibiting the degradation of extracellular matrix. Extracellular matrix is due to the urokinase. So this is the results which we obtained, that most of the, or you can say the almost all the molecules goes and bind to the same site. And of course, the best molecule we have selected, that is a ganoderic acid, which confirms that it goes and bind to particular active site of urokinase and it inhibits. We use the active Autodoc also for analyze the kinetics of this, and we try to find out the inhibitory constant also, and calculate the inhibitory constant, how this enzyme urokinase is inhibited by the these molecules. So So if I try to summarize this, that we started with the chemoinformatics, where we tried to find out the molecular property of these molecules and confirm that they are following the Lipinski rules. Then move forward with the another chemoinformatics tool, where we confirm that yes, they are going to follow the ADME property and they are quite safe. And then we move to the in vitro lab, that is on the bench. We we tried to study the anti-proliferative activity. We tried to find out the selective index, and we were excited to see the selective. So it is having the very highly selective for the cancer cell. So we move to the cell culture, cell cycle analysis, which also confirmed that yes, cells are not multiplying now as efficiently as earlier they were multiplying. Cells are, very few cells are in the replicating phase or synthesis phase. And then we try to understand the cell death by apoptosis, confirm the apoptosis. And then again, with the help of literature, we try to find out the possible target, which has given us an indication of the migration to decide the migration assay, confirmation of migration assay. We again moved to the in silico and did our docking experiments, and we confirm that yes, this may be the mechanism, or we are almost sure that this is the mechanism by which the ethanolic extract of fruiting bodies as well as the mycelial extract inhibit the cell growth of cancer cells. So I will just making a statement that the integrated approach for validation of traditional and herbal medicine is the need of our, and we have to design the experiment in such a way that outcomes should come and so as a proof. So it will give a confidence to accept these herbal drugs for treatment as well as the prevention of cancer. And as I said, that these are not toxic compounds, their side effect will be less, as well as if we start integrating this with the modern medicine, the concentration or doses of chemotherapy can be reduced, so the suffering of cancer patient will be reduced. So something like this we are play in our laboratory. Second part which we do is we optimize the growth conditions and production of this, which I'm not here discussing. But as we all know that these labs are not developed overnight. Many people are there who are supported this. So this is the team which has played a very important role in developing this lab, like Dr. Varsha Vadagaonkar, Dr. Kanchan Bhagwat, who was my first student, PhD student, who successfully grown the roots in the reactors, Dr. Sarika, Jay, Prachi, and this work which I am presenting is from the Harshada Pille's work and Mr. Roshan. And as lot of money is required, so almost the financial support was from the Science and Technology Cell, Government of Maharashtra, University Grant Commission, New Delhi, DRDO. Of course, the lab is supported by DST, FEAST also. DBT has generously provided a funds for bioinformatics, setting up bioinformatics lab, CSR for the fellowships, and yes, technical support, which always I get from the RL Jammu, that is the lab for the secondary metabolites and plant metabolites, I consider, in terms of especially the bioprocess as well as the biochemistry part. And NCCS is an institute which has already always supported us when I was a student from that time, or when I was working in the industry from that time. I'm associated with NCCS when the Dr. Wag was a director, and the actor at Mumbai for the cell line support. So this is all about the work which we carry out in our lab. And I hope that I have not taken much time and completed my assignment on time. So It's all. Thank you. Thank you very much. And I will be glad to answer if there are a few questions.
Thank you very much, sir, for your excellent and informative talk. Any questions from audience, please? Yes. Sir, I have a question from audience. इकडे ये. तू त्यालाच बोलू दे.
Nice to hear you, sir, and thank you, sir. My question is there any running research to produce new variety of edible mushroom?
In my lab, no. In my lab, no. We are not working on developing a new mushroom or those. My lab is dedicated for the validation of this herbal molecules using the cell lines. We are integrating the cell lines research with the plant research. This is a one part, and secondly, if there is a system is ready and if you want to take it to in vitro system, then yes, we try to develop a process for optimization of these products by taking it to the root organ culture or suspension culture or some other approach.
Okay, thank you.
Thank you.
Thank you very much, sir, and thank you. I would like to request Professor A.P. Pathak ma'am to conclude the talk.
Good afternoon, sir. So it is great pleasure for me to offer vote of thanks to great researcher, Professor Wadegaonkar sir. Respected Wadegaonkar sir is playing very vital role in various activities of School of Life Sciences, and I really feel proud to have my PhD referee here as a speaker. I sincerely express deep thanks for delivering a talk on bioactive biochemicals from Ganoderma species against crucial targets of cancer management. It was really a wonderful experience with you, sir. Thank you for giving us time. Thank you very much, sir.
Thank you. Thank you, ma'am. Thank you very much.
Thank you very much, sir. Now, moving forwards for the valedictory function. Soon we will start the valedictory ceremony. In 5 minutes, we will start the valedictory. After valedictory, there is a photo session with guests. So please be there for photo session. And main gate of SLS, we will have photo session with chief guest at outside of the hall.
जय भीम सर काय ना कॉन्फरन्स चालू नाही इकडे हो 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 चालतंय
आम्ही अचूकपणे टेस्ट करू. म्हणजे ती एक प्रकारची मल्टीपर्पज लॅब असेल. त्या मल्टीपर्पज लॅबमध्ये विविध प्रकारचे DNA आणि RNAचं ॲनालिसिस आणि बाकीचे व्हायरस डिटेक्शन ह्या सगळ्या गोष्टी आम्ही त्या ठिकाणी करू. आणि दुसरी मोठी गोष्ट म्हणजे आताची लॅब आहे ते लॅब मी म्हणेन की सेमी ॲटोमेटेड आहे. येणाऱ्या काळामध्ये ही लॅब फुल्ली ॲटोमेशन करण्याचा आमचा उद्देश असेल, जेणेकरून टेस्टिंगचा कालावधी पण कमी होईल आणि ॲडिशनल फॅसिलिटीसुद्धा त्या ठिकाणी मिळतील.
मला अतिशय अभिमान आहे की अशा या अडचणीच्या आणि आव्हानात्मक काळामध्ये स्वामी रामानंद तीर्थ मराठवाडा विद्यापीठ म्हणजेच नांदेड इथे असलेलं आपलं विद्यापीठ यांनी पुढाकार घेतला आणि नांदेडला टेस्टिंग सेंटर ज्याला केंद्र सरकारच्या संस्थेची मान्यता आहे अशी सेंटर हे नांदेडच्या विद्यापीठामध्ये स्वामी रामानंद तीर्थ मराठवाडा विद्यापीठामध्ये सुरू झालं आणि त्याच्यामुळे त्याचा फायदा एवढाच झाला की तातडीने टेस्ट होऊ लागल्या. नांदेड, परभणी आणि हिंगोली 3 जिल्ह्यासाठी असलेलं हे केंद्र आणि त्यातून लोकांना फार दिलासा मिळाल्याचं निश्चित आहे.
तर आम्हाला समाधानाचं वाटतं की आपण या सगळ्या लोकांचे प्राण वाचवण्यास आपण थोडंफार आपल्या पद्धतीने कॉन्ट्रीब्यूट करू शकलो. हे सगळे विद्यापीठ असो, नॅशनल लॅब असो, याचा सोशल इम्पॅक्ट असलाच पाहिजे. ही सोशल रिस्पॉन्सिबिलिटी जर सगळ्यांनी हे केली तर मला वाटतं हे जे काय येणारे नवीन संकट आहेत ते संकटाला आपण यशस्वीपणे सामोरं जाऊ शकतो आणि यातनं आपण सही सलामत बाहेर येऊ शकतो.
कोरोनावर मात करण्यासाठी आपले ध्येयवादी नायक रस्त्यावर, हॉस्पिटलमध्ये आणि प्रयोगशाळांमध्ये अहोरात्र शौर्याने लढत आहेत.
Welcome to all delegates and dignitaries. Welcome, sir. Now we are in a last session of this 3 days international conference on advances on bioactive molecules, ABM 2022. To improve the golden moment of opportunity and catch the good that is in our reach is the great art of life. A very good afternoon to invited guests, professors, students, ladies and gentlemen. I am Dr. Amruta Kanakarande. It is a matter of immense pride to stand before all of you on this session. On behalf of the institution and on behalf of the organizing committee of ABM, we are delighted to offer a gracious welcome to all of you. To give a red carpet welcome to everyone assembled over here and who joined us virtually also. Welcome all. I would like to welcome Dean Sir of our university, Dr. L.M. Wagmare Sir. As a president, we have with us Dr. Giri Sir from NCL as a guest. We have with us. Welcome, sir. We have with us our cheerful Director of School of Life Sciences, Professor S.P. Chauhan Sir. Welcome, sir. And we have with us Dr. A.P. Pathak ma'am, convener of this international conference. Welcome all. I would like to welcome all the committee members and I would like to request Dr. T.A. Kadam sir to please welcome of today's president, Dean sir, Dr. L.M. Wagmare sir of our university, Dean Science and Technology of SRTM University, Nanded. Sir, please. By offering a book. I would like to request Dr. B.S. Survese sir to please welcome Dr. Giri sir. Book. this occasion. Sir, please.
So we are here for the concluding session of this long discussions, scientific discussions and deliberations during this 3 days international conference. Advances in bioactive molecules. Actually, School of Life Sciences got very little time of 6 months, less than that, for the preparations and confirmation of guests and various speakers, and that's why this conference was little bit a hybrid mode. Major sessions we conducted offline, but around 30-40% were the guests from various countries, like we got from Germany as well as from Thailand, and it was a little bit online mode also. So anyway, so this is a conference in the AC hall, but we are planning for a next conference, which will be our workshop model of a meeting of all delegates on field only. It may be ecological or ornithological, or basically it will be in zoology. Okay, so be ready for that, as you are here from since last 3 days with your great patience to listen various things from biomolecules and various complex things like the drug designing and all that from various researchers all over India as well as from abroad. On this occasion, I congratulate our head of the department microbiology, Dr. A.P. Pathak Madam, for leading this conference as convener, and my colleagues from our department, Dr. Survase sir, Dr. T.A. Kadam, Bhosle Madam, for continuous remain operated to conduct these all sessions here. Unfortunately, for some office duties, I could not attend all sessions in this conference, and all the delegates coming over here. On this occasion of this valedictory function, we have today's guest, the Dean, Faculty of Science and Technology, Dr. L.M. Wagmare sir, the chief guest of honor for this function, Dr. Giri Ashok from NCL Pune, the Pathak Madam, the convener of this conference, my friend for this conference, this function, Dr. Deepak Sate, in University Administration, then Dr. Ambadas Kadam also here, and all my students and the online guest we called for the success of this conference. So I personally feel okay. This we tried in short time, and I hope all of the students, basically it is for students, PhD researchers, you got some new ideas to proceed your research or to plan your further research in this line of work. There are thousands of opinions, thousands of lines of research, biomolecule, bioactive molecules and drug deliveries and all that, molecular aspects as well as some field aspects you may come across during this whole tenure of your education, either in a PG or PhD. So more events are coming. Nearly COVID pandemic has stopped, we consider, and there will not be further online all drama. I think it will end, and we will all of you enjoy in coming days to come more conference and more scientific events. So thank you very much for your, I again appreciate for your patience. Major students remain in the hall to listen. Thank you.
Thank you very much for your words, sir. Now, for feedback, I would like to request Dr. Dilip Dudmal sir, who is from Dindori College, Nashik. Sir, please.
Hello, everyone. Greetings from Nashik district of Maharashtra. This is Mr. Dilip Haribau Dudmal, an assistant professor working in the Department of Zoology as head of the department, MVP's ACS College, Dindori, Nasik. Well, I'm here to propose or share some feedback from the deliberations which were happened over the last 3 years. 3 days. I'm sorry. So at the very outset, let me tell you that the time will not allow me to give feedback of all the sessions that has happened over the last 3 days. But I must tell you, this is the first scientific gathering that I am attending. Ignore that. This is the first scientific gathering that I'm attending, and I hope that you guys are also attending after 2 days of 2 years of havoc making pandemic. And this is actually an opportunity for all of us to share, to learn, to meet our people of all the scientific fraternity who are working in the area of bioactive components or the molecules. I want to thank the organizers for selecting this particular theme for this program, that is advances in bioactive molecules, that world over the scientists and researchers are working to solve the problems of before humankind. Talking about the resource persons, I will not take a particular name because all the resource persons were actually they were a feast for all of us, the developing brains, including the MSc students as well from the School of Life Sciences of this university. All the sessions were nicely planned and they were actually executed well. And the food, actually, I must appreciate the food as well. The food was delicious. And lastly, I must thank all for the hospitality, for giving me the opportunity to meet my friends, to meet my fellow professors. And I'm surely, I can promise you that I'm going to take a lot from this conference. Thank you for this opportunity to share my feedback. Thank you.
Thank you very much, sir. Now I would like to request Dr. Suyash Kathale from Bharati Vidyapit, Pune. Sir, please.
Thank you so much, everyone. So from last 3 days, every day was very nicely planned, and so much of bombarding of scientific knowledge and information was there every day. So there are take-home message from it, and everyone will definitely have a nice career out of it. It will help you in your MSc dissertation and in your PhD. I would like to thank all the organizing committee of this conference, all the teaching and non-teaching staff, especially the volunteers are pay a lot of efforts in completing this conference. And I would like to congratulate all the coordinators and of this conference for successful completion of this conference. Thank you so much.
Thank you, Dr. Suyash. Now I would like to request, is Maithili online?
Hi, yes, ma'am,
Anybody?
I'm online. Hello? Hello? Hello?
Yes, yes, yes.
Hello, ma'am. Am I audible?
Yes, yes, yes. You're audible. Please, you can. Yes, Maithili, you can start your video, please.
Okay. Hello, ma'am. Actually, ma'am, I am in campus only. Actually, 3 days session was there, and I was continuously feeling like why I am not there. This was my mistake, ma'am. Actually, I should be present there. I am virtually attending because of some reasons. We have some responsibilities of attending sessions here also. So I was managing all the things. So Dr. Giri sir has said that mass spectroscopy is a very wonderful technique, so that we can uh, the number of metabolites we can sort out from there. So continuous 3 sessions were there, very wonderfully organized, and we got as per previously they have spoken that bombardment of sessions were there. So science lectures we are bombarded, sir, and we will work like a mass spectrometer, and we will explore the scientific knowledge to whole world. So senior said eminent scientists delegates are there, and this was my first international conference. And we will first paper presentation I have also done. I don't know how it was, but SRT university has given us such a nice wonderful platform, so that whatever it is we have presented our paper. So this was the biggest satisfaction of this conference. And Giri sir, young PhD scholars are there like me. So we will, from all of us, I promise you, sir, we will contribute for our science faculty, that we will explore like mass spectrometer. You have given us a very wonderful thought, sir. Thank you, sir. And thank you, SRT University.
Thank you, Maithili. Thank you for your feedback, and surely in future you will be here physically in our next international conference soon. So thank you very much for your feedback. Anybody is interested again to give a feedback? Please welcome. From MSc students. Yes, please come. Your good name? Your good name? Dhage Priyanka is giving feedback. Yes, welcome.
Good evening, everyone. Myself, Dhage Priyanka from MSc first year, microbiology. And I'm here to share my thoughts about this international conference. Actually, I'm slightly nervous right now, so my voice is trembling. But until now, before this, I was a student of Dr. Babasaheb Ambedkar University, and my previous college was Yogeshwari from Ambajogai. And then I joined here, and I didn't know that international conferences are like this. And even this conference halls, I feel so much proud that I'm a student of this university now. And I try to gain knowledge as much as possible, and be one of the proud student of this university, that our professors will be proud of us, and to gain and to give as much as possible to this university and to this science. Thank you.
Thank you, Priyanka. Now, again, anybody's interested? So, okay, thank you. Now, the next most awaited moment for every student's life. Talent and skills are the crops that are groomed and nurtured and given to all possible opportunities to grow and bloom. This is a much awaited event in every student's life, as it gives the returns of the efforts put in by them throughout their academic endeavors. Now, it is a moment with full of joy and celebration. For this moment, I would like to call Hemalata Bhosle ma'am to please announce the winners for oral paper and poster presentations. Ma'am, please.
So good afternoon, everyone. I'm here to declare the names of participants who have given oral presentation either in offline or online mode, even for poster presentations in offline and online mode. So I will take the names of participants one by one. First, for oral presentation in online mode. So the first prize goes to Akib Nisar from Biochemical Sciences Division, Bharati Vidyapit, Pune. Second prize for online oral goes to Maithili Kesraikar, Department of Biotechnology, Gulbarga University, Karnataka. Congratulations, Maithili. Are you there now?
Yes, ma'am, I'm online only. Thank you, ma'am.
Okay. Yes. And the third online oral prize goes to Joita Kar from Department of Biotechnology, Pachunga University, Mizoram. So now the oral winners of the oral presentation in offline mode. First prize goes to Pushpa Karare, School of Pharmacy, SRTM University, Nanded. I request Dean sir to give the honor. Second prize in offline oral goes to Rihana Beg, School of Life Sciences, SRTM University. So I request Surose sir on behalf of her to accept her letter of appreciation. And the third prize for oral presentation in offline mode goes to Swati Jadhav, School of Life Sciences, this university. So she is a PhD student of Professor Kamble sir. Congratulations to all the winners of oral presentation. Now it's time for participants who are declared as winners for poster presentation in offline mode. First prize goes to Rutika Hatkar. She is a student of School of Life Sciences. Second prize for offline poster presentation goes to Abhiruchi Deshmukh, Department of Biotechnology and Bioinformatics, MGM College, Nanded. Third prize for offline poster goes to Divya Chauhan, again from Department of Biotechnology and Bioinformatics, MGM College, Nanded. So now it's time for those participants who won poster presentation in online mode. Because number of participants who gave their online presentations for poster were few, only 2 winners we declared. First prize goes to Rucha Godbole, Department of Botany, Savitribai Phule, Pune University, Pune. And second prize for online poster goes to Ankita Karnawat, Department of Zoology, Dayanand Science College, Latur. So congratulations to all the winners.
Many congratulations to Ankita. Thank you all dignitaries on the dais. Thank you. Now I would like to request Dr. L.M. Wagmare sir, Dean of our University, Science and Technology. Like to request, please felicitate Dr. Giri sir. Sir, please. Thank you very much, sir. Thank you very much, ma'am. And now I would like to request Dr. Giri sir to please say a few words on this occasion. Sorry, one announcement, please. I would like to request Professor Pathak madam to please say a few words as a closing remark on this occasion, ma'am, please.
Thank you, Amruta. So, respected dignitaries on the dais, Honorable President Wagmare sir, Dean Faculty of Science, Honorable Chief Guest Ashok Giri sir, respected Director Chauhan sir, I will be more happy if my colleague and co-convener, respected Kadam sir, respected Survasi sir, and organizing secretary, Professor Kamble sir, Professor Bhosle madam, and Zhore sir, if join here and share this stage. Okay. Respected dignitaries on the dais and off the dais, especially Sate sir, Ampadas Kadam sir, and Jogdan sir. All participants, students, and directors of campus schools, teachers, and supporting staff of School of Life Sciences, research scholars, those have helped us lot. Before offering a formal closing remark, I pay tribute to late Professor Khobragade sir, great researcher, humble teacher, and pioneer of ABM 2022. And now I offer this closing remark. So on behalf of organizing committee ABM 2022, I am delighted to deliver this closing remark on success of International Conference on Advances in Bioactive Molecules 2022, which have been organized by School of Life Sciences, Swami Ramananda Tirth, Marathwada University, Nanded. The conference and events were held in hybrid mode due to extraordinary situation generated by COVID-19. The committee successfully hosted events as plenary and invited talk with oral and poster presentation in online and offline mode without any technical fault, and therefore I congratulate all committee members. I believe that during these 3 days, we have all had insightful interactive discussions and great chance to share outcome of research. Even virtual participants were also very active. And therefore, let me present a brief valuable information regarding the success story of ABM 2022. So the day and duration of conference is 7 to 9 April. Working hours really amazing, 9:30 a.m. to 8:30 p.m. Unbelievable. However, the mode was hybrid. Offline mode have been joined by 50% participant in very impressive Senate Hall of this university. For online mode, committee members have generated a Zoom link and event is also live streamlined with YouTube. more than 3 resource persons have joined overseas, and more than 20 dynamic resource persons have delivered their talk and discussed their research in bioactive molecule. Amongst these, the 3 speakers, those were from different countries. They have delivered very informative information and shared their research with us. Directors of various research institutes, eminent professors, talented pharmacologists, and scientists have delivered nice talk and elaborated their successful journey in world of bioactive molecule for us. Keynote speaker, Honorable Om Kumar sir, have illustrated on bioactive molecule for Alzheimer's disease. Honorable Baban Ingole sir have explored marine world for bioactive molecule. Guest of honor, Honorable Yogesh Sauche sir have illustrated role of human microbiome in bioactive molecule research. Honorable Dr. Rakesh Baurya sir, in spite of poor health, have nicely explained the traditional sources for bioactive molecule. Honorable Upadhyay sir have focused on treatment for TB, of course, with bioactive molecule. I am really very happy and pleased to brief the impressive role of female scientist speaker in this conference. Honorable Dr. Amarja Joshi ma'am have enlightened alkali files and their applications in bioactive molecule. Honorable Professor Anita Patil madam have nicely explained anti-malarial activity of bioactive molecule. Honorable Professor Peshwe ma'am have nicely explained on microbial enzymes, extraction, characterization, and very, very useful cultivation techniques, especially useful for microbiology student and researchers. Honorable Professor Sarma sir have elaborated role of rare herbs in treatment of many disorders. Honorable Sutawat Benjakul sir from Thailand have illustrated collagen-based product. Honorable Pillai sir, again overseas, have opened and discussed on drug discovery and its journey. Honorable Professor Arun Kharat sir have explained on phytochemicals. Honorable Professor Shivraj Nile sir, our alumni actually, have illustrated about cancer treatment. Again, it was a proud moment for our school. Professor Barkate sir explored biosimilar and biologic product. Honorable Professor Sonone sir have explained asparaginase in chemotherapy. Honorable Ashok Giri sir, Honorable Chatterjee sir, Honorable Venkatraman sir have explained area of their research very nicely. Honorable Selvin sir, very cooperative professor from Pondicherry, have delivered talk on synthetic biology. And Honorable Ade sir, Vadegauskar sir have delivered talk in last session. The response received from all participants was very encouraging from beginning itself. So total more than 100 participants were there, more than 60 abstracts and near about 30 full-length paper. Although this is the last day of conference, the process is not over yet. There are still some steps that should be done. Please fill feedback form from using link uploaded on Telegram. First, I express sincere thanks for submitting manuscripts and abstracts with us. And then I wish to share something. We have already submitted your manuscripts to care listed journal. Editor may communicate to corresponding author. Kindly reciprocate that message. As per declared policy, best 20 papers will be published in care listed journal without any processing charges. For non-selected papers also, we offer you to publish it as book chapter with ISBN number, paying very nominal charges. Editor has to select papers, and therefore we kindly ask your patience here. Last, however, very encouraging announcement. I am really happy to announce that we have selected, I think this part already we have covered, that is best oral and poster presentation. I congratulate all winners. Personally, I thank everybody without taking anybody's name, because remember, this is the success story of team, and the role of participant is very, very crucial. So on behalf of we all, I really express deep thanks to all of you, to participants, those who have joined here in offline mode and online mode. Thank you very much.
Thank you very much, ma'am. Thank you for your. Thank you very much, ma'am, for your kind words. Thank you. I would like to request Dr. Giri sir to say a few words on this occasion. Sir, please.
So probably I think whenever I get a time, I have to come and talk. So very nice to have. So respected Varmari sir, Kadam sir, Madam, Patak madam, and all my friends who are here, and most importantly my students. It was very nice that yesterday I came and interacted with several of you, and today as well. This very important conference. I mean, madam, and all of you mentioned that it was most bombarding. I don't think you should say bombarding. That's probably not so good word. Maybe we should say that you have molecular feast or feast of in real science that you enjoyed. I'm sure you will carry a lot of interesting messages. Few things I would like to tell you that look, life is beautiful. Okay, you need to take it in a right spirit. Your future is basically depend upon your present, what you do. You take some lessons from your past, but most important in my opinion is you should enjoy present. You should be asking questions. Actually, in our philosophy, we always keep saying that we should have self-assessment. In Marathi, we called it as a Sihavalokon. So that's good, but then I feel that present is most important. A lot of friends are my friends ask me, how are you today? And I say, better than yesterday. So they always have two questions. One is, what happened yesterday? I said, nothing. It was good, but my today is better than yesterday. So that today what I was there, tomorrow, I mean, yesterday what I was there, it's better than yesterday. That means today I'm doing very well. And then one very curious friend asked me, that means you are improving every day. I said, yes, that's the life. Yeah. So I would like to share a few things with you. That one of the problem with the student coming from our area is a kind of, I will say, we get really less exposure, but thank you all this team who organized such conference. This is opportunity for us, especially for students to interact with the people of a different kind, but we did not have much opportunities. But don't keep that as a hesitation. I could have been very happy to see that there are several people who would like to give the feedback about conference or ask the question. So this is most important. I will tell you, 2012, I was in Weizmann Institute of Science as a Dr. C.V. Raman Fellow, and they asked me to give a talk. And on second or third slides, there were 6 questions. So I think that habit, I mean, I don't know what kind of a hesitations you guys have, but remove that. Actually, the good speakers or good audience will never say any question is a stupid question. Question is question. I think generally it is treated as a question. So don't have that kind of hesitation. Unless you speak up, you talk, you are not going to give develop yourself, speak, because today, whatever good science we do, to some extent, we should be a good articulate. You should be able to really present in a nice way, so that you can convince your story to your colleagues or your students or your friends. So this most important, I think, keep that habit. So also I would like to share a few things. The way that if I compare the students from the Western world towards our world, and then maybe the students of our universities, like whether it is Dr. Babasaheb Ambedkar Maharashtra University or several state universities, that it's like I will say that we have a potential. We have a researcher. There are a few things which are very, very important. One is actually everybody has to be intelligent. I don't think, that's my personal opinion, that people are not intelligent. Everybody is intelligent. Sometimes scores or academic scores are okay. Sometimes, you might have less score. That doesn't mean that you are not intelligent. But for research, there are two more qualities which are really important. One of them is patience. Research is not in a goody-goody. So if you plan 10 experiments or 8 experiments doesn't work the way you want. That's one thing. And the second thing is there is no any excuse for hard work. I say it is a hard work because people call it as a hard work. And I will tell you about this hard work. One of my senior colleague came and said that, why you are working so hard? You should spend some family time with your family and so on. So I said, is there any problem that did any member of my family complain to you that I don't give a time? And second thing is, why you say it is a hard work? Because I enjoy this work. So it's not any more hard work, but for saying that the people call it is hard work, but you have to work. Unless you work, you cannot produce a data. And for a good researcher, good researcher is always a researcher who keeps the record in a proper way. Because till your BSc, I know even I did the same thing, that how we write our notebooks once in a year or once the exams are coming. So we sit together as a group and then we write the notebook. But that's not the correct way of doing research. Because generally I learned from my seniors and my teachers that record keeping is most important. If anybody of you are coming to Pune, I will show you my notebooks from the day I joined PhD. Since then, even what happened today, it is recorded in the notebook. And this habit I learned from my teacher. And this really helped because I feel that is my property. I have around maybe 250 notebooks. And I feel these are my property, because you will find everything, like what I was doing on the first day, or maybe on the during my PhD, whether even I was traveling to my native place is recorded. So it's sometimes good to become a nostalgic and go back and what you are seeing. And more importantly, at the same day, I used to conclude, because a lot of time, some of you might be researchers, and a lot of time I also get the answer from my students. There is a negative data, or experiment didn't work, or some problem in the experiment. So I feel, and I would like to give this message based on my own experience, that there is nothing like a negative data or positive data. Because based on whatever the literature search you do, and based on the hypothesis that you put forth, you come up with some kind of, this is how it should work. And when you carry out the experiment, you get a result. And once you analyze the result, if technically the results are very sound, you should forget your hypothesis, because you become biased to your hypothesis. But results are telling you something very important. And you should start believing on results. And once you do that, you can really, I will say, discover something new. Because in my opinion, and with all these senior people, I would say that if your hypothesis work, it is a good, I will say, good data for research. And if your experiment or your hypothesis did not work and your experiments are solid, technically sound, and if you can explain these results, that becomes a search. So that's the difference. So while doing research, why we do research? To find something. So research is basically, the name itself is research. But while doing research, there are only, I will say, 5 to 10% possibilities that you can discover something new. And most of the time, scientists, researchers forget to explain what we have got. And I will tell you one story. And this is about bioactive molecules in plant. So in 2001 or 2000 sometime, scientists wanted to express some of the gene for color in petunia. This is the chalcone synthase gene. So they wanted to have intense color to the petunia flowers, so that it has a, and this research was done in the Netherlands, because Amsterdam is very famous for flower market. And they wanted to increase the intensity of the that color, pink color in petunia. And they overexpress the gene which is responsible for this color and compounds. And what they found is that instead of having a dark color, they see the patches which are much more lighter or maybe even white on the corolla or on the petals. And they couldn't explain what was the story. I mean, of course, it was they published something, but they did not really dig out what exactly, why this has been happening. In 2006, Milo's group was working on C. elegans, which is another model system. And they also got similar data with some other gene that something is not working as per their hypothesis, but they digged out what exactly the problem is. And they found there is something called RNAi, which is called RNA silencing. And that got Nobel Prize in 2006. So please understand, sometimes results keep telling you something, but then if you ignore it, you will miss the boat. So whatever, I mean, everybody might not get Nobel Prize, but then if you go back and see, then you can explain the results very nicely. When I joined NCL, I was also very scared. Probably you guys will be in the same situation when you go to NCL. Everybody is with in-shirt and shoes and so on. I was so scared. I thought this is not a place for me. I wanted to run away from that. But somehow I came for an interview, so I have attended the interview, and interview was very nice. I think morning you saw, I show you Professor Ranjekar's picture in this family, PMB family picture. So he was, as the head of the department, he was also there in the meeting, the interview committee. And towards the end, he asked me, Ashok, your interview was good, but I want to understand what is your weakness and what is your strength. So I was not able to speak good English because, in our area, if you start speaking English with your colleague, you are outcast. That's a problem. So after my even PhD, I was not very good in speaking English as well as in writing English. So I said, sir, weakness first. I will tell you weakness, but of course I was not so good and so in speaking, but I said, yeah, I cannot write good English as well as I cannot even speak good English. And that's what something must have been said. And then strength was, I said that from Aurangabad, I did PhD in 4 years and I published 5 papers. One of them is in phytochemistry and so on. So what he said, and this was very important for all of you, he said, we are not worried about your grammar as well as glamour. We like your content. And this is what people will always appreciate. So if you are a good worker, if you have put some effort, I was not so smart, as I said that I wanted to run. And then he came outside once my interview was actually completed, and he said, and he put, I will say, his hand on my back, and he said, we don't want to lose like researchers or scientists like you. I thought I should jump from that floor because this person is saying me I'm a scientist. I never considered this a scientist. So please believe on yourself. I mean, you really do not have to get worried. I will just speak for two or three minutes and I will finish. So I will tell you that everybody has the story. I mean, all of us come from a very different background. So my parents never went to the school. So my first dream was to have a Pitachi Chakki in my village because people have to travel walk 3 kilometers to Dalnasati. guys. Then my second dream, when I was in 9th or 10th standard, somehow I got into the social welfare, Samaj Kalyanchi Shaळaमध्ये Lowulla. So something I convinced my parents, so they sent me there. It was very interesting. So my fees was for lodging, boarding, जेवण, शाळेची फीस आणि राहण्याची फीस ₹5 महिना. Being supported by the Samaj Kalyan, social welfare. So somehow they thought, okay, let him go to the high school, and that's how I went. And they gave me a target: don't take too much stress. You pass 10th standard in 3 attempts. So in first attempt, you take all the subjects except math and English. I'm sure you must be witnessing that. And then in October batch, you take English and later on maths. But somehow I passed in first attempt with a good mark. So this is a story about most of you. I mean, maybe now situations are much better because, I mean, now the internet and all these facilities are there. But what I'm trying to say that You cannot really have people like us coming from these rural areas. We cannot have a career plans, and we don't do this IQs and so on. I mean, whatever comes as an opportunity, we take in a very positive sense, and we throw ourselves in that. And that's what I called it as a ownership. So whatever you do, it doesn't matter what you do. Whatever you do, you should take it as that it is mine. And you have to really believe on yourself and work. I think success will come to you. I never ever thought in my life that I will get a position in a NCL as a scientist. Okay, but I can tell you today, friend Gajanan told me that last week, there was a top 90 scientists in the India in the area of biology and biochemistry, and I'm one of them from India. So I don't want to take this as my career. This happened because of my students, my mentors, my colleagues. It's not like this all 175 papers we published, whether it is in science or nature. It was all with my, I will say, very hardworking and sincere students, my great mentors at respective place in my career, as well as my colleagues who has helped. So I was counting how many people I have published these 175 papers, and it number came out something like 350 different co-authors in these 175 papers. So collaborations are must. We cannot really work in isolation, particularly the people like us, where we don't have really all great facilities and infrastructure. We should really complement with each other and not actually compete. If we compete from our region, it is as good as we are society. We will not progress unless we collaborate, unless we understand. And also another thing is, no area is less area. I gave 2 days back conference in, keynote address in angiosperm taxonomy conference. And I was wondering what I'm going to talk, because I'm not a taxonomist, neither I'm a botanist. But then they appreciated my talk very much, because they talked about plant taxonomy, and I was talking about molecular diversity in the plants. So how these plants are so interesting and so much diverse, and how this molecular information is helping taxonomists to really categorize the plants. And you must have seen some of my slides that how it happens. So believe on yourself. I think success, as I repeat, the success will come to you, and never really give up. I have learned more from my mistakes than my success. So success is very easy, and that you get it, but you don't learn. But if you fail, if you really struggle, I think you can make a lot of wonders in your life. Last example I will give you of my own, just because to tell you that how this happens. So when I got a job as a research associate in NCL, sir, the problem was decided by a guide, which typically happens. So guides come up with the problem, and the problem was that how plant defends. And then for system that I was given was chickpea, that is harbara, chana, and insect called pod borer. And I worked for almost 3 years for that, and nothing worked as we expected. So the first experiment was that if chickpea is highly susceptible to the insect, what you are going to identify? Because insects are enjoying, that means they are overcome the defense mechanism. So I thought I am coming from a village background. I'm really smart, because most of the studies which are reported that there is defense molecule present in chickpeas from mature seeds. That means the seeds what we get in the market. And I said, I am a farmer's son. I know that insect doesn't eat mature seeds. It eats developing seeds. So I collaborated with Mahatma Phule Krishi Vidyapit Rahuri and tagged thousands of flower and collected developing seeds of chickpea, starting from pollination like 6 days, 12 days, like that. So till the seeds mature. And what we found after all this analysis, it took almost 7 to 8 months to collect the seeds and analyze. And what we found, the most susceptible stage, which is a mid-mature stage, which we called it as a 36 days after flowering. That means what we eat chickpea as a raw or as a dhala khato apan na to. And that has the highest number of these defense molecules. Still, that is the best for the insect as well. So it was a completely, as per my hypothesis, it was a negative data. That means the stage which is having highest number of these protease inhibitors that we are working on, and the insect is actually eating and enjoying that. So it was complete, I will say, upside down, in the sense it was very difficult for me to accept this 1 year work and there's the negative data, so called, as per the hypothesis. Then I thought maybe most of the studies people are working with a constitutive defense, because there is something called constitutive, and when there is a damage, plant produces something more. So there also we could see that plant is actually increasing the accumulation of these protease inhibitors. That was probably still a good data, but it was not, I mean, the plants were susceptible. Then I thought most of the people, and that's what we do, we purchase something from Sigma or something from company, and then we use this as a trypsin as a molecule. I thought I don't want to do that. We started dissecting the insect and isolating the protease activities from the insect midgut, and there we got answer. And that paper we published in Plant Physiology, several of you know, and that changed my career. So after publishing that paper from my postdoc work, I got in one day 3 offers, one from Netherlands, another from US, and third from France. So one paper, you don't need 10 papers. If one good paper, and this paper, I will be very proud to say you that this paper is cited more than 220 times till this point. So that's the strength of research, and that's the strength of data. So I really would like to encourage you, take your careers in a very positive mode. Don't go into the like what will happen, whether you will get a job. Nobody will tell you. I mean, even our prime minister decides that they will give you a job, it is not possible. So job should come to you, and it will come to you. I promise you, job will come to you. For example, I mean, all these senior faculties, we know we don't get a good student, neither we get good faculties if you want to really. So that means for good people, there is always a job. And I mean, I will say, just believe on yourself. I would like to thank all of you, all of the organizers, for giving me this opportunity to share my thought. I wish you best. And anytime if you are coming to Pune, please visit NCL. I will be very happy to show you or interact with my students if I'm busy, and share what we are doing. Thank you very much. Jai Hind, Jai Maharashtra.
To lab and say our results are getting or not. So sometimes we researchers said that no sir, results are not there and all that. So every time means once in a week is मतलब वो होता ही था, रहता ही था. So later on, sir said, sir, everybody is saying that ki, no, sir is continuously saying, no results are negative, no results are negative. Your perception is different. You have to read again and again. So something is really productive come out of that. And sir is also saying that. So thank you very much, sir. Thank you for your words. Now.
So with kind permission of the chairman of this session, and I'm here to say something. As Giri sir said, yes, we have some weaknesses and some strengths. So our main weakness is we fail, or our failure to express or demonstrate our strength. And we do have, our life sciences do have many strengths, but many times we are not able to express, we are not able to demonstrate it at the right platform. And I would like to take this opportunity to introduce to you our heroes from our life sciences, our COVID warrior. They are sitting here. Can you guys please stand up, all the COVID warriors? Please have a big round of applause for those people. Sir, these are our heroes, and since from last 2 years, these guys are working in the lab day and night. Kodgiri madam, please. Khaja sir is there? Yeah. Yeah. So these are the people, they are working day and night since from last 2 years, and they have set an example before the country, all the universities of the country, that we can use our resources for the human welfare. And they are doing it. They were not joined for some sort of benefit or some monetary gain, but as a service to society, they have joined, and still last 2 years they are working. And this is very proud moment for all of us, life sciences. And I think this is the right platform to appreciate their dedication, their support. So thank you very much for allowing me to introduce to you our heroes. Thank you all.
Thank you, sir. Now I would like to request Dr. Wagmare sir, for Dean of our university, for presidential address. Sir, please.
Honorable Dias, Dr. Giri sir, scientist from NCL, Dr. Chavan sir, Dr. Patrick madam, honorable members of the Dias, Dr. Jogdan sir, Dr. Amadas Kadam sir, Dr. Chatte sir, Kadam sir, Zhore sir, Suryavanshi sir, other dignitaries of the Dias, and my dear participants and student friends. It gives me immense pleasure to be the part of this August gathering. We are at the concluding session of 2 days international conference on, 3 days international conference on advances of bioactive moleculars. I really congratulate the coordinators, director, head of the department, faculty members, students, supporting staff of the school for initiating the activity and completing it in a very successful manner. I hope during these 3 days, you have successfully observed, successfully achieved the objectives which have been set before initiating the activity, before launching the conference. The conference has been organized in 2 modes. It was hybrid mode, that is offline, online. We should look always after the advantages of online mode also. We have seen the disadvantages of learning through the online mode, but we should look after the benefits of online mode. Because this was through offline and online mode, many faculties, many experts, many participants, many students from international, abroad, foreign universities have joined this conference and they have presented their research. I personally feel that the postgraduate students of this university, this school, must have been benefited a lot from this conference. Research scholars, they are doing research, but you are going to start your research. Now you are basically in the first phase of taking the research ahead. So I'm sure that you must be benefited out of this conference. Most importantly, that you must have a networking with all the researchers, networking with all the experts, networking with all your faculty members. It should not be for a short duration of time. It should be for a long duration of time. I once again congratulate you all for participating in the conference. I also congratulate those who have won the different prizes for presentation, oral presentation, poster presentation in offline mode, online mode. I also congratulate all the faculty members, all the staff members of the school for taking this conference in a very successful manner, Thank you very much. Thank you.
Thank you very much, sir. Thank you. Now I would like to request Dr. L.H. Kamble, sir, to propose a vote of thanks on this occasion. Sir, please.
सर्वांना नमस्कार. मागच्या 3 दिवसापासून आपण इथे बसलेले आहात. आजही तेवढेच चेहरे हसते दिसत आहेत जेवढे पहिल्या दिवशी होते. हे पाहून आम्हाला जो हुरूप येतो ना तो प्रचंड आहे. आणि कॉन्फरन्सचं नाव ॲडव्हान्सेस याच्यासाठीच असतं की प्रत्येक विद्यार्थ्यांनी थोडं तरी ॲडव्हान्स व्हावं, एक स्टेप पुढे जावं. एवढे मोठे मोठे सायंटिस्ट आपल्या सोबत आले. त्यांनी त्यांचे विचार, त्यांचा रिसर्च, त्यांच्या लॅब, त्यांचं इन्फ्रास्ट्रक्चर, त्यांचा वेळ तुमच्यासाठी खर्च केला. एरवी तुम्ही जर ट्राय केले ना त्यांच्या इन्स्टिट्यूटला जाऊन तुम्हाला त्यांच्या गेटमध्ये सुद्धा एंट्री भेटणार नाही. आज ते मोकळ्या वातावरणात इथे येऊन तुमच्यासोबत चहा पीत बसले, जेवत बसले. ह्या गोष्टीचा ऍडव्हान्टेज किती घेतलं मला माहिती नाही, पण येणाऱ्या भविष्यामध्ये जरूर हे शिका की ते तुमच्यासाठी इथं आलेले आहेत आणि त्या वेळेचा पुरेपूर फायदा घ्या. वेळ परत येत नाही. तर एवढं बोलून let us come to the official program of saying thanks. So I would like to thank each and everyone who is associated with online, offline, in every mode, with the support that you see that man, Umesh Duldaj, Dr. Umesh Duldaj. He is sitting right behind there, the computer since last 3 days. Not only 3 days, but he is working since last 8 or 10 days non-stop to make this conference a success. He has compiled this book with the great help of our Pathak madam and Bhosle madam, that they are constantly editing and reporting the authors that okay, these are the changes we need. आणि जे काही फीडबॅक्स मिळाले त्या फीडबॅक मध्ये बरेचसे रिजेक्ट झाले, बरेचसे याच्यातही हे आलेले आहेत. त्या सर्व एफर्ट साठी उमेश, थँक्यू व्हेरी मच. आपण खूप साऱ्या कमिट्या बनवल्या, त्याच्यामध्ये फूड आहे, रजिस्ट्रेशन आहे, ट्रान्सपोर्ट आहे. संजय चव्हाण मागचे 3 दिवस झाले रात्रंदिवस रेल्वे स्टेशन जातो येतो, गेस्टला सोडतो आणतो आणि ही इज वर्किंग राऊंड द क्लॉक. त्याच्यामुळे संजय चव्हाण तुमचे पण धन्यवाद. भागवत लाड त्याची कंपनी, म्हणजे यांच्यासोबत ह्या पुस्तकामध्ये जेवढे नावं छापलेले आहेत, एक एक घेत बसले तर बाहेर अंधार होईल, आपल्याला ग्रुप फोटो पण काढायचा आहे. त्याच्यामुळे असं गृहीत धरा की मी ह्या सर्वांचे नावं घेतलेत. ह्या सर्वांचे धन्यवाद. आणि अमृता कुठे गेली? अमृता, द गर्ल बिहाइंड सक्सेस ऑफ दिस प्रोग्राम. अमृता पहिल्या दिवसापासून आतापर्यंत तशीच उभी आहे, तशीच चार्मिंग, तशीच हसत आहे. नो स्ट्रेस, नो टेन्शन. म्हणजे हे जे वर्क कल्चर आहे ना, हे शिकण्याचा प्रयत्न करा. अजिबात थकायचं नाही, काम करत राहायचं. ओके. तर आपल्या सर्वांना झाडबुके सर बाहेर आहे, लाईट कमी होत आहे, आपल्याला ग्रुप फोटो काढायचा आहे. सर्वांनी फ्रंट गेटच्या समोर, इथं नको, इथं होणारच नाही, पटकन जाऊ. जेवढ्या लवकर जातील तेवढ्या लवकर जायचं. पायऱ्यांवरती एक पायरी सोडून उभे राहायचं आणि असे एकमेकांना ब्लॉक करायचं नाही. बरोबर मध्ये चेहरे घेऊन उभे राहा. जे उंच आहेत ते दूर जा, जे लहान आहेत ते खाली राहा. ओके. आणि फक्त थँक्यू म्हणून कोणाचेही थँक्यू किंवा आभार मानता येत नसतात. आपण त्यांच्या ऋणामध्ये कायम स्वरूपी राहावं असं मला वाटतं. एवढं बोलतो माझे 2 शब्द संपवतो. फ्रंट